Good morning, and welcome to our current workshop with the Global Food Systems Initiative at Kansas State University entitled Food Safety, Current Research and Future Needs of Meat Safety. I'm Dr. Maureen Olevnik, coordinator of the Global Food Systems Initiative here at Kansas State University, and I'm really proud to present to you the first and hopefully a series of workshops in the area of food safety. We will be focusing on very critical work that's being done from the research groups on campus here at K-State. And I'm really happy to say that we've gotten some good outside presenter here today that's going to be giving us an overview from a governmental perspective. A couple of housekeeping issues we've got here. You would have gotten a message on how to log into this workshop. There was also information there on a Qualtrics connection that will allow you to ask questions. As those questions come in, I will monitor those, and at the end of each of the presentations, I will ask those questions to the presenter. So we're not going to have direct one-on-one. So if you want to have anything specifically asked, please just fill it in the Qualtrics question survey, and I will be glad to offer those questions up to the presenter. If for any reason we run out of time or we're not able to proceed with that, I will make sure that we capture all of the questions and get them to the presenters, and they can get back to you in good time. I believe with that, the next step is for me to introduce Dr. Peter Dorhout, the Vice President of Research here at Kansas State University. And Dr. Dorhout is going to do the introductions and welcome.
Good morning, everyone. And if you'll bear with me, I'll take a moment to share a screen. Wouldn't be a Dorhout introduction if there wasn't a PowerPoint presentation. I want to thank everybody for joining today, and hopefully you can see the slide on the screen. Maureen, is that the case?
Yes, we do. Thank you.
All right, thanks. I want to start at kind of the beginning, I guess. Kansas State University's had a long legacy, really, since it began as a land-grant university in 1863. And some of our earliest alumni were instrumental in advancing food safety and food science, plant science, agriculture, and ultimately had a hand in the development of the U.S. Department of Agriculture. While many recall that the Land Grant Act was first introduced to Congress on May 5th in 1862, there are very few who remember, and perhaps those who have worked with USDA will recall, that it was only 10 days later that the same President Lincoln created the U.S. Department of Agriculture. And it is those two legacies of Abraham Lincoln that have tracked in parallel these many years. So I want to introduce you to two of our alumni, David Fairchild and Charles Marlatt. They clearly overlapped during their time here at Kansas State, and I wanted to just briefly share a little bit about each of their contributions. David Fairchild was very much interested in helping to diversify the food plants here in the United States at the turn of the 19th century and into the early 20th century. Charles Marlatt, who studied here and became a faculty member for a few years before joining the USDA, was very much focused on the insects that traveled with plants. And so he and David Fairchild actually had a bit of a love-hate relationship when it came to importing plants into the United States. That was David's goal, and Charles was very much interested in protecting the plants that we had here in the U.S. David worked with Barbara Lathrop, who was a philanthropist and very interested in helping David advance the plants and plant varieties here in the United States, and recognized that it was critical for the strength of U.S. agriculture to diversify the food that was being grown here across the diversity of land and climates that we had here in the United States. Charles was, as I mentioned earlier, on the other side of this equation, looking at developing regulations that guided the kinds of plants and ultimately the insects that were being imported in the United States in order to protect our agriculture. He was almost singularly responsible for the Plant Quarantine Act of 1912 that ultimately led into what we now know as, or have come to know as, the Agricultural Research Services at USDA. And from that, we saw an evolution at USDA of the Animal Plant Health Inspection Services, ultimately the Food Safety and Quality Services, that have become our keynote speakers' home organization within the Food Safety Inspection Services in the USDA. So K-State's had and its alumni have had a long history in getting us to where we are today in terms of thinking about food safety, food security, and ultimately what we're going to be doing in order to feed the 10 billion people of the planet in just a few decades. Let's advance that slide. Other aspects of K-State's legacy in food safety and food security have been in the inventions and disclosures that have come out of K-State. The first disclosure was actually a stopper for bottles stoppers for bottles that were used in packaging. And the first patent application was a plastic container for food freezing capabilities. You can go on and see the long list of patents and licenses that were garnered by K-State over its early history, and the impact that it's had in food safety and food security. And of course, it's been that legacy that has helped create the future home of the National Bio and Agro Defense Facility here in Manhattan, Kansas, which anchors the animal health corridor that is one of the largest concentrations of animal health companies in the world today. And it's a great pleasure to be part of that legacy and to invite our speakers today to share a little bit about the research and the emphasis that they're placing on food safety and food security, in particular related to disease. And so it's my pleasure to introduce Dr. Melanie Abley, who will be our keynote speaker for the day. I'll stop sharing my screen and get back to the main page. Dr. Abley is currently the Deputy Director of the Risk Management Innovations staff, or RMIS, within the Office of Policy and Program Development at Food Safety Inspection Service at USDA. Prior to her current position, she was a senior microbiologist with RIMS. She started out in the Office of Field Operations and in the Food Safety Inspection Services as an enforcement investigation and analysis officer in 2013. She joined the USDA and the ARS as a postdoctoral researcher after completing her PhD at the Ohio State University, which is today celebrating its 150th anniversary as a joiner of the land grant legacy here in the United States. Her dissertation focused on salmonella and Campylobacter prevalence and molecular characterization in cattle and swine within the farm to fork continuum. And before I let Dr. Abley speak, I'll say I had a personal interaction with Campylobacter a number of years ago, back when I was actually working in the former Soviet Union. And when I first heard the name, the first thing that came to mind was Velociraptor. And believe me, there were a lot of commonalities between the feeling of a velociraptor inside of me and the effects of Campylobacter. So thank you, Dr. Abley, for your research, and really appreciate the time you're taking to share with us today, and look forward to your presentation.
Thank you for that introduction. Thankfully, I have not had to experience Campylobacter to my knowledge, or salmonellosis, so that's a plus. So that was a great introduction to my agency. So I work for the Food Safety Inspection Service within USDA, and we are primarily responsible for the food safety, proper labeling, wholesomeness of meat, poultry, and processed eggs that are processed in the United States. And recently, meat also includes fish of the order Siluriformis, or catfish. So all other food follows under FDA. So I am going to talk about a few different things. I kind of put together a presentation based on what I thought would be really good information for you all to have. So the first piece I'm going to talk about is the new technology process within my agency, and how we approve new technologies to be used on meat, poultry, and processed eggs products. I'm also going to talk about this up and coming new kind of technology that's out there, cultured animal cell food products. So I'd like to go over just a little bit of what the framework that we have set out for that will be, as well as giving you some updates on some of our performance standards and non-0157 sampling expansion, so you can kind of get a feel for what is coming next as far as the regulatory piece. I also want to go over and help you find where we have our FSIS research priorities, and then also kind of rounding it out with just a plug for NIFA and funding opportunities. So within the US, within FSIS, how do we define a new technology? We consider a new technology anything different than what is currently being used to produce a meat or poultry product for consumers. The agency has a regulatory interest in a new technology if it could affect product safety, inspection procedures, inspection program personnel safety, or if it would require a waiver of a regulation. So this all kind of falls under our net of new technology. It is essential in the public interest that the health and welfare of consumers be protected by assuring that meat and meat food products distributed to them are wholesome, not adulterated, and properly marked, labeled, and packaged. So our ingredient submissions make up a large percentage of our new technology submissions, and they are reviewed via our FDA FSIS joint review process. And we have a specific MOU that details out those specifics. Within this joint collaboration that we do with the FDA is determining the safety of whatever the new GRAS ingredient is, whereas FSIS determines the suitability of the use of that ingredient within FSIS regulated products. And I'll go into a little bit more detail in my next slides on what that review entails and what we're actually looking at. And so you can see on the slide, there's a link to the MOU and the number, so you can see that, and just kind of lays out what our different roles are in that process. And so how it normally works is that whenever there's a company that would like to list their ingredient as GRAS, they would first go to the FDA, they would submit their submission to the FDA, and then once the FDA has received it and they see that it has something to do with any of the products that we regulate, they would then share that information with us and we would start our review at the same time. So here is just a general outline of the phases of the review process within FSIS. So companies first submit scientific or technical information that describes the technology, whether it be a new method, a process, or a procedure, and what the technology is intended to accomplish, as well as the beneficial or adverse effects that the technology is expected to have on FSIS regulated product. Each new technology submission is reviewed by a multidisciplinary team that is tasked with evaluating the scientific or technical support. The review team members are selected based on expertise and are from varying staff, including our Office of Field Operations, our Office of Public Health Science, Office of Policy and Program Development, which is the staff that I'm involved with, Office of Management, and Office of Data Integration and Food Protection. In most cases, the new technology reviews results in a no objection letter. So we've reviewed it and we found that the data was sufficient. And so we have no objection to that being used on our product. However, there are other outcomes that vary depending on what the submitter has requested. Our current review process is inherently flexible in that it is designed to provide for industry innovation through the development of new procedures, equipment, or processing techniques. FSIS considers a new technology anything different than what is currently being used to produce a meat or poultry product, going back to our definition. So the suitability piece relates to the effectiveness of the process or substance, in this case, the production of agricultural products from cell cultures in performing the intended purpose of use and the assurance that the conditions of the use in an FSIS regulated establishment will not result in an adulterated product or one that misleads customers. The suitability data supports that neither the intended use nor the substance would adversely affect the wholesomeness of the product. This may include data that illustrates how the technology affects the organoleptic properties, such as taste, color, odor, and texture of the product. The submitter also provides support for all of their claims, and the technical review team reviews that support. And based on the results of the technical review team, a no objection letter determination could be made. So the suitability data, again, supports that the intended use, that neither the intended use nor the substance would adversely affect the wholesomeness of the product, illustrates the effectiveness of the ingredient or substance, assures the conditions of use in an FSIS regulated establishment will not result in an adulterated product or one that misleads consumers, and illustrates how the technology affects the organoleptic properties. So an example recently that I can give is that for whether it would result in a product that might mislead consumers, this hasn't been approved yet. So we're still kind of trying to figure it out. But there's a company that has thought about using red blood cells, dehydrated red blood cells, and adding them in from cows and adding them into ground beef prior to high pressure processing, HPP. And their claim is that typical HPP will cause kind of a graying of the meat and is not as appetizing to the consumer. And so it's less likely that a consumer may want to buy meat that's been high pressure pasteurized, even though that may be a safer product overall. So they thought that if they added these red blood cells to the meat would look more red after the process. So we internally are struggling with the idea if there's a chance that that red coloring could mislead a consumer to think that when that product does meet its end shelf life, is it still going to be organoleptically consumable? Is it still going to be good? And so so that's just one really short example that I have of that particular piece that we're looking at with not misleading the consumers. So here is a little bit more detail on what the technical review team consists of and what their duties are within the technical review team. You can see that, you know, our Office of Field Operations includes both members from headquarters and specifically the district office, or if there's an establishment involved, then there'll be somebody that's at the establishment that can provide us information directly related to what's going on there. And so they assess whether the technology presents any enforcement issues or interferes with any inspection activities. We also have our Office of Public Health Science, which assesses the adequacy of the scientific information, the proposed study or protocols and methods used to ensure that the conclusions that are drawn are scientifically accurate. We also have our Office of Policy and Program Development. So we're the managers of the technology process, and we determine if the technology meets our regulatory requirements. And then our Office of Management is ensuring that the safe and healthy working conditions are part of the whole process and determine whether the technology might impinge on our labor management agreement. So specifically looking at our inspectors that are in plant every day. Could this technology have any effect on them? One example is peracetic acid. Sometimes that forms a gas that can affect the lungs of those around. So if somebody is proposing to use that at a certain concentration and there isn't adequate exhaust or anything else, then that could affect the health of our inspectors that have to be on the line. And then lastly, the Office of Planning, Analysis, and Risk Management, OPARM, and they determine whether the statistical methods are included and whether they're statistically valid. So that is our kind of really robust review team that is looking at all of these new technologies that come in. This is a timeline, general timeline of what we expect to take for our new technologies. At any point, we could be, the timeline could be skewed if we get a submission that isn't complete and we need to go back to the submitter and ask them to provide us more data. Sometimes they didn't realize they needed to provide us certain data and they actually have to go off and do a whole nother study. And so, you know, depending on what the ask is and what's going on, this timeline can be augmented. But this is our general, what we hope to accomplish. And we hope to have them out in about 60 days. We have resources on our website, and also the FSIS Directive 7120.1 is revised hopefully every month, every couple of months, with any new submissions that we have approved. That way we keep it kind of up to date. And the information that we have on our website, which I'll show you in a couple more slides, our lookup table has all of the ingredients or antimicrobial interventions that we've approved since the ones that have been in our regulations. So our first substances that were recognized as safe and suitable are included in our regulations in 9 CFR 424.21C. And so that lists many of the ones that were very initially used. And then over time, it was decided that we would, in order to add new substances, we would have to conduct rulemaking to change our Code of Federal Regulations. And so we changed our process, and now any of the new substances are just updated in our directive. So we kind of have two different systems to see what's been approved. This is the lookup table that we have on our website, and it has a lot of really great searching capabilities. So you can put in a particular substance, a particular intended use. And this is the information that we try to provide when we do approve a substance, so that there's parameters around it, so that establishments know how the product was intended to be used, on what products and at what concentration, if that's important, a dwell time, whatever the really specific information is that the establishments should need to know will be included on our website. And we do tell establishments that this listing the 7120 is not their support for using the antimicrobial intervention in their process. They still have to provide scientific peer-reviewed papers or have conducted their own validation study with sampling to show that the particular parameters that they've chosen for their unique establishment make the most sense. Here's kind of just a listing of the different groups that we have within our safe and suitable ingredients. So you can see kind of the various different areas that we're approving in. And also just is just another way to kind of break down specifically with the antimicrobials, the ones that we've kind of approved over time, CPC, PAA, organic acids. So these are the ones that are kind of most commonly used in our establishments. Here are just some additional web resources that you can go to. We have a guideline that is provided for establishments or chemical companies or anybody that wants to put in for a new technology. It provides guidance on what we're looking for to help have a good submission starting out, so there's not as much back and forth. Also our directive, and then a link to the 9 CFR again. And also, I didn't talk a lot about our waiver program, but also kind of within the whole new technology umbrella, we do list out waivers that we've granted to certain regulations to allow establishments to innovate or to change something that wouldn't necessarily be allowed under our current regulations. But if there's enough support or enough information that we can see that after the waiver, they've provided us enough information to show that it's not going to adversely affect the food safety of the product, then we do have some waivers out there. And this is a link to those waivers on our website. And then there's also on our website, there's additional related information. The next piece is I just want to briefly go over some of the roles that we've kind of defined for this new area of the cultured animal cell, human and animal food products. So the information I'll be describing today is primarily drawn from the March 2019 formal agreement that was made between FDA and the USDA regarding the oversight of human food produced using animal cell technology derived from cell lines of USDA amenable species. I'll provide a link to the agreement at the end of the presentation, and the key points are that oversight will depend on what species of cell the cells are from and whether they're being used for human or animal food. So FDA and FSIS will divide responsibility for oversight of human foods incorporating cultured animal cells derived from livestock, cattle, swine, sheep, goats, and siluriformis fish, and poultry, which include chicken, turkeys, ducks, geese, guineas, ratites, or emus and ostriches, and squab. FDA will provide oversight for human foods incorporating cultured animal cells derived from fish, other fish, and seafood, and the FDA will provide oversight for animal foods regardless of the source of animal cells. So together, we haven't seen any of this come to market yet. This is still all theoretical new technology. So when we see our first one, we'll develop a very detailed framework for the shared oversight at harvest. And we're defining harvest as the point where the cell lines are no longer just growing and reproducing and forming whatever product that's going to be, but they've actually kind of stopped that initial growing phase and have moved into the now it's kind of the end product that needs to be further processed, cut, informed whatever that next step would be. So that's kind of our defining line between what FDA will handle and then when FSIS would take over the process. We would also develop a joint process for identifying and implementing any needed statutory or regulatory authorities. We'll collaborate both from the headquarters and in the field at the establishments. We'll also develop joint principles for product labeling and labeling claims and work together to investigate any food safety issues. Here are some contact information if this is a direction that any of the research is going to go at Kansas State University. We are encouraging companies or researchers or anybody to bring us in early in the process so we can really kind of help figure out what the regulatory piece is going to look like for these products. It's important, you know, both from the regulatory understanding as well as informing technical and business strategy. So the contacts are listed here specifically for this area. At FSIS, it's Matthew Michael. And then for FDA CIFSTAN, there's the email, and then also for the FDA CVM. So those would be for food for animals would be CVM. And then we've also just provided some other further resources that we have in this developing area. We did have a public meeting with the FDA and the formal agreement, as well as a Food Safety Magazine article. So this is just a little bit of information about that large area that's sometimes a little bit of a black box. So the next phase in my talk is to talk about some of the performance standards that are going to be coming out. So for beef performance standards, we're using the salmonella data from our sampling and the risk assessments we have developed, and have proposed a new salmonella performance standard for beef manufacturing trimmings. We published the proposed standards on October 28th of 2019, requesting comments, and posted the risk assessment that we used to develop the standards. Under the Healthy People 2020 goals, FSI is committed, along with the CDC and FDA, to a 25% reduction in annual cases of salmonellosis. The Healthy People 2030 goals have come out with the same percent reduction goal for salmonellosis, so our proposed standards are still consistent with the 2030 goals. Using published results from the CDC, FSI estimates a median of about 80,000 annual cases of salmonellosis associated with consumption of cuts and intact beef and ground beef contaminated with salmonella. We estimate that approximately 53% of these illnesses are associated with ground beef. Thus, to meet the 25% reduction goal, there would need to be 10,600 fewer annual illnesses caused by raw ground beef contaminated with salmonella. We use this data to develop the standards. The proposed standards would apply to establishments producing more than 50,000 pounds of ground beef or beef manufacturing trimmings per day. This approach would account for approximately 91% of the total raw ground beef and 96% of the total beef manufacturing trimmings production volume annually. And as we explained in the risk assessment, the approach would also focus agency resources on raw ground beef establishments shown to have the highest salmonella prevalence. In the Federal Register notice, we explained that we intended to use a 52-week moving window of FSIS sampling results. This is similar to what we do in poultry to assess whether establishments meet the performance standards. Approximately 1 year after the new standards are made final, the agency plans to post information on the FSIS website on the individual establishment as either meeting or not meeting the pathogen reduction performance standard. We also announced we would need 48 samples to be able to assess whether establishments are meeting the standards. We also announced that we intend to increase salmonella sampling to once per week in beef establishments that produce greater than 50,000 pounds of raw ground beef and beef manufacturing trimmings per day, so we can collect those 48 samples. Although we opted not to propose pathogen reduction performance standards for raw ground beef and/or beef manufacturing trimmers in lower volume establishments, FSIS will, however, continue co-analyzing for salmonella in all samples it collects for STEC analysis from these establishments to monitor ongoing pathogen prevalence. For ground beef, the standards would apply to about 75 establishments. And we predict that approximately 18% of establishments, or about 14, would initially not meet the performance standard based on our previous sampling history. Once implemented, if approximately half of the establishments producing more than 50,000 pounds of raw ground beef per day, not meeting the proposed performance standards, subsequently began to meet the standard, this would result in a 25% reduction in salmonella illnesses from that product. Consistent with current procedures, FSIS will continue collecting and analyzing raw beef samples for applicable STEC, depending on the product and salmonella. We received 87 comments on the proposed standards. They were from consumer groups, industry groups, law firms, other countries, and individuals. And the comments were on the risk assessment, alternative approaches to salmonella control, and how to approach the measurement of salmonella control. So we hope to be able to go out with that final rule soon. It's in the queue. So moving on to Campylobacter in poultry. We are currently assessing whether poultry establishments meet the salmonella standards for young chicken and turkey carcasses, comminuted chicken and turkey, and chicken parts. We are posting information on each of these establishments and providing detail as to whether an establishment is meeting or not meeting the standards currently. However, we also establish Campylobacter standards for poultry product, but these Campylobacter standards all need to be updated because of a change in our testing method. We changed from direct plating to an enrichment method, which is much more sensitive. While we are co-analyzing all poultry products for both salmonella and Campylobacter, we are in the process of updating the Campylobacter standards. This is important because Campylobacter is the most common bacterial cause of foodborne illness in the United States, and most non-dairy Campylobacter illnesses are attributed to the consumption of poultry. We proposed updated Campylobacter standards for comminuted chicken and comminuted turkey products in August of 2019. The revised performance standards account for the method change from direct plating to enrichment and were established to provide the same public health benefits and industry costs as the original standards. We were able to propose the Campylobacter standards for comminuted chicken and turkey products sooner because sample collection does not involve the use of a new neutralizing buffer peptone water and collection of rinsate. This allowed us to use the enrichment generated data collected previously for these products. Thus, we have sufficient data to be able to develop the proposed updated standards for comminuted product, but did not have enough Campylobacter data to propose new standards for carcasses and parts. We are collecting data for poultry carcasses and chicken parts using the enrichment method and intend to propose revised performance standards for those product categories in the Federal Register. So we're still working on that one. This is a summary of the proposed new standards for comminuted products. To meet the standard, the establishment can have no more than 5 positive FSIS samples out of 52, or 9.6% positive if fewer samples. And FSIS would need 11 samples to be able to determine whether the establishment meets the standard. This is consistent with how the agency categorizes poultry establishments for salmonella. Once the agency starts assessing whether establishments meet the Campylobacter standards for comminuted product, FSIS would categorize them as category 1, 2, or 3. Category 1 would be establishments that have achieved 50% or less of the maximum allowable percent positive in the standard using the most recently completed 52-week moving window. Category 2 would be establishments that meet the standard but have results greater than 50% of the maximum allowable percent positive during the most recently completed 52-week window. And lastly, category 3 would be establishments that do not meet the standard. The standard would apply to 481 establishments. In case, because there are more positives for salmonella and Campylobacter in poultry compared to salmonella in beef or pork, we took a different approach to establishing the standards. We plan to apply the standards to the higher volume establishments. For the poultry establishments, the standards apply to all product, unless the establishments produce less than 1,000 pounds per day. Also for beef, because the standards allow for so few positives, the establishment would be considered as either meeting or not meeting the standard. We would not set up 3 categories for beef. So there are some differences. And a lot of that has to do with the percent positive that we find in these establishments and Historically, in beef, the largest percent positive of salmonella seems to be in the larger establishments, whereas for poultry, it's kind of the opposite. So that's why there's differences in the way that we set up our standards. We hope we are providing strong incentives for establishments to meet the standards. Moving on to pork. The HACCP regulations included salmonella pathogen reduction performance standards for market hogs. Since that final rule and until 2011, FSI has conducted verification testing in market hogs for salmonella and assessed establishments against the performance standard. This was discontinued in 2011 after years of sampling revealed consistently low salmonella recovery rates across the industry. In addition, a market hog baseline survey conducted from August 2010 to August 2011 further supported the finding that salmonella prevalence on market hog carcasses was too low to justify ongoing sampling. FSI has estimated the prevalence of salmonella in market hogs was 1.66% on the carcass. For those reasons, the final rule on swine slaughter modernization eliminated the pathogen performance standards for market hogs. Instead of keeping those standards, we decided to focus resources on sampling raw pork cuts and comminuted pork product. We plan to propose performance standards for comminuted pork and pork cuts based on this data from our baseline sampling. We started the exploratory sampling in 2015, and we have been sampling at both slaughter and processing establishments. We also plan to publish a risk assessment to support the standards. We think a performance standard is necessary for pork products because salmonella outbreaks in pork products continue to occur. Among FSI's regulated product, the highest percentage of salmonella outbreaks are associated with poultry, then pork, then beef. Non-FSIS, 64% salmonella illnesses is for non-FSIS, and then poultry is 18%, pork 11%, and beef 7%. As shown on this slide, the largest establishments account for most of the pork production. We found that salmonella is higher at these establishments as well, similar to what we found in beef. So we increased sampling at the larger establishments. We also plan to propose that the performance standards would apply to those larger establishments. In addition, because of the low recovery of STEC in samples tested in our pork sampling, FSIS is no longer analyzing samples from slaughter establishments for STEC. Rather, we are sharing sample enrichments from USDA's ARS for further analysis of STEC recovery in a collaborative study. For establishments that produce greater than 6,000 pounds of comminuted product per day, we will collect samples 5 times per month. The establishments that produce greater than 50,000 pounds of pork cuts, we also collect 5 samples per month. For all other establishments, we will continue to sample several times per year for a total of 500 samples at those establishments. We updated our instructions to inspectors to increase sampling and to inform them of these changes in FSIS Notice 4119 and hope to have the proposed standards out by the end of this calendar year. We still hope to have that though. So beef and the expansion of non-0157 STEC testing and a little background. On June 4th, FSI has proposed to expand non-0157H7 STEC verification sampling and analysis to all raw beef products that we currently test for E. coli 0157H7. In response to outbreaks, FSI has announced in 1994 that E. coli 0157H7 adulterates raw non-intact beef. We later clarified that non-intact beef intended for use in non-intact product is also adulterated when contaminated with E. coli 0157H7. So for instance, if there are trimmings that are considered intact because they haven't been blade tenderized or otherwise the inside hasn't been made non-intact, but they're destined for ground beef or something else, they are considered to be tested and would be adulterated with STEC. It's the same for primals and subprimals that are destined to be made into non-intact steaks as well. In 2011, FSIS announced in the Federal Register that raw non-intact beef products such as ground beef as well as raw intact beef products that are intended for use in raw non-intact products are adulterated if contaminated with the 6 specific non-0157 STEC serogroups listed on the slide. In June 2012, the agency began verification testing for these serogroups in beef manufacturing trimmings. Currently, FSIS does not test ground beef, bench trim, or other raw ground beef components for non-0157 STEC. However, FSIS tests ground beef and those other components for E. coli 0157H7. We only test beef manufacturing trimmings from non-0157 STEC and E. coli 0157H7 currently right now. So we intend to expand our non-0157 STEC testing to all beef products that we currently sample for 0157H7 because of outbreaks and to prevent contamination. Some examples of outbreaks are listed on this slide. Also, based on our beef manufacturing trimmings, STEC testing results, E. coli 0157H7 and non-0157 STEC are not typically found in the same sample. This further supports the expansion of the non-0157 STEC testing. This expanded sampling would apply to applicable imported raw beef product and to ground beef that FSIS tests at retail. Generally, controls that are validated to address 0157 are also effective against non-0157 STEC. Therefore, if establishments test for 0157H7, we generally consider that to be sufficient verification testing for the non-0157 STECs, unless the establishment or FSIS has found product positive for non-0157 STECs. As we have stated in a POSTIC and publicly available Ask FSIS Q&A and in FSIS Directive 10,010.2, in response to one or more non-0157 STEC positives, establishments must ensure any additional testing conducted includes non-0157 STEC as part of their validation, verification, or reassessment requirements, and in their supporting documentation, so that establishments are able to demonstrate control over these pathogens. The next phase of my presentation is to go over our research priorities, and these are found on our FSIS website, and we update them yearly. If anyone was registered for IAFP, there is a poster that I pulled a lot of this information from, but in addition, the poster also includes our proposed research studies, which are on our website yet. So the ones I'm going over now can be found on our website. And the way that we come up with our research priorities is just internally, as all of us that are scientists are working on various initiatives within the agency, if we realize that when we're developing our guidelines or our performance standards or any of our other documents, if there's research gaps and there's not, or even if we get asked FSIS questions from establishments that say, I can't find any scientific support to support this process that I want to do, we kind of look at that and then come up with as an agency whether we want to put that forward as a research priority. So, FSIS, we are really a regulatory agency. As you've kind of seen from my various previous slides, we take a lot of samples. We do a lot of testing, but our testing is verification testing. It's really not used for research. However, we do provide that information publicly so research institutions can look at our data and use it for research, but we aren't funded to do research. Within the USDA, that would be the Agricultural Research Service, which they do look at our research priorities and try to incorporate those in their performance plans. And then also we work with NIFA, which is my last slide is opportunities within NIFA to talk about our research priorities. So they're aware of what we kind of need as an agency. So the next couple slides just kind of go over what our current research priorities are in the different categories. So we have 4 different categories, and the first one is chemicals of potential concern. So you can see various different pieces of that and what we were interested in. And then on our website, if you click on each of the priorities, it will give a more detailed explanation of our thought process and what we're kind of like, what angle we're going at. And if there's ever a question about any of our research priorities and you want to reach out and ask for clarification or to figure out more of a background or anything, feel free to reach out and ask us those questions. We would be happy to help kind of clarify that for you. So the next category is biological hazards. So different screening, detection, enumeration methods, or pathogen characterization. You can kind of see what the various different research titles could be for some of those projects that we're looking at. And this is biological hazards continued. Intervention strategies. So these involve different antimicrobial interventions, or if there's any, so one of our new research priorities is around, are there any synergistic or even not as good interaction between using different antimicrobial interventions throughout the multi-hurdle process? So if chlorine is used here and then the very next spray is PAA and then the very next spray is CPC, are there any interactions between those chemicals that could be more beneficial at producing a log reduction, or on the alternative side, are they maybe producing off-gassing or anything that we may not be interested in? So we really haven't seen that work done. So that's going to be a new one that's going to come on our website soon. We also have one under animal welfare and another one under label verification. So our labeling claims, you know, if they're saying that this is beef, is this really beef? Or any of their other various claims, as well as allergens too. So I also just wanted to put up there, in case anyone wasn't aware, we have a very extensive culture collection that we save and freeze back. So if there's ever a time where anybody would like any of our isolates from outbreaks, or, you know, with particular antimicrobial resistance profiles, or, you know, just about anything, we probably have an isolate because we have so many isolates. So we have we have a way to share those. So if interested, there's a way that you can get more information about that on our website. And the last page, the last piece that I just wanted to part with is, of course, funding, which unfortunately FSIS doesn't provide, but within the USDA, we have NIFA, and now they're soon will be located in Kansas City once everyone goes back to the office. So they have, you know, I'm sure most of you are aware, but they do have this AFRI grant that you can reach out to and see if there's any information that fits any of the work that you're doing at Kansas State. So I just wanted to kind of end with that, and now I'll take any questions that may have come.
Thank you so much, Melanie. That was great overview of the processes and programs that you've got in place now. One of the questions that I've got here is in relation to, you do an awful lot of testing, clearly, and have tons of data, lots and lots of information. What are the expectations with modeling and with artificial intelligence and those types of things in the programs that you're managing?
So I would say that within FSIS, we're not currently utilizing any of the artificial intelligence within our data. I know that there are groups within FSIS that are looking at new tools, specifically around the kind of salmonella serotype virulence era. Like, are there some salmonella that could potentially be considered more virulent or more able to cause disease in humans? And so we are kind of looking at artificial intelligence to possibly mine some of the whole genome sequencing data with external collaborators. But within FSIS, we're not currently utilizing that.
do you, in the fact that you're not utilizing it now, is it something that you would be looking to have as a tool in the future?
So I would say, generally speaking, we in the agency are very open to new technologies, new ways of doing things. So yes, I think that there are some ways that we could utilize that in the future, given, you know, with being in the government, it sometimes is challenging to get those new technologies in initially, but if we definitely find the need, and we can put out a contract and be able to utilize that. Yeah, I could see it being utilized in the future for sure.
Another question that we have is around the idea of the microbiome, and what is the focus from your agency's perspective on the microbiome and how it may impact, in the general sense, the safety of meat?
Right. So to just kind of like provide context, I think one thing is that for us as a regulatory agency, we really need the isolate in order to be able to show one way or the other that isolate was in this particular food and could be utilized in the future in an outbreak investigation or something else. And so for us, regulatory speaking, it's hard for us to envision how we could really utilize the biome information for us specifically. However, we're scientists, we understand that shifting the microbiome can have huge implications for whether, it's an effective place for pathogens or not effective for pathogens. And so I think that for us, that would probably fall more on maybe the pre-harvest side, which unfortunately, we at FSIS, our jurisdiction starts once the animal reaches the doors at the slaughter establishment. And so anything that happens on the farm, that is regulated by our sister agency, the Animal Plant and Health Agency, APHIS. So we can't really control what happens at pre-harvest. We do put out guidelines, and we do talk about, various different probiotics or prebiotics or different things that the science, the research has shown could be helpful in reducing salmonella or campylobacter or STEC or whatever the pathogen might be in the animal. But I think from a regulatory standpoint, it's hard for us to envision a future where we don't actually have the isolate to be able to have the regulatory authority to make the decisions that we do about adulteration or illness outbreaks and things like that. I hope that answers your question.
I think so. Here's one other. On this last slide, you have funding opportunities. Are there any specific areas that that you can point to where more focus should be paid from a university perspective on areas? Is that making any sense at all? That I think the question is, do you have any specific ideas on some of these grant activities that you would suggest that we stay focused on?
Well, I would say from just a, since we're the Food Safety Inspection Service, from a food safety perspective, the food safety, nutrition, and health part of AFRI would be probably where, if you were wanting to do any of the research priorities from our website or anything that we saw as gaps, that would be probably the arm that would make the most sense to go for. But since I'm not with NIFA, I can't really speak for NIFA, but I know that they have a lot of outreach and things where they can provide a lot more information and direction for the best places to apply and how best to apply for those areas.
Great. Okay, we've got another one that just came in with salmonella testing, particularly in beef. Does FSIS have any indication or plans for collecting information related to the role of lymph glands in commuting product contamination?
Right. So, we're currently working on a guidance document for beef, and we are going to provide some information from the scientific literature on salmonella contamination in lymph nodes. We know that it's possible. We know that it's there, but at this time, we don't have kind of the big enough evidence to be able to instruct establishments that they need to remove those lymph nodes before they can be used in ground or otherwise non-intact products. However, we are looking at, and we have looked at some of the data, because there are some establishments that do remove some of the larger lymph nodes, because those have to be removed as part of AMS requirements for meat that goes into the school lunch program. So we do have some information about that, but there will be more information that comes in our guidance document that we are putting together currently for beef and salmonella, which we hope to hopefully have out by the end of next year.
Great. Do we have any other questions from the audience? We've got a little bit of time here. Is there any follow-up that you might have, Melanie, on on things that we should be thinking about from a research perspective on campus?
I'm looking forward to the next presentations to see what you guys are doing. I think that I plan to stay on, so if there are any additional questions that come up later, I can be available. But no, I think overall I provided the context of our proposed performance standards, because I think that that's going to be the direction that a lot of industry is going to go to try and come up with new interventions or new strategies to help so they can meet those performance standards. So that's why I wanted to bring that to everyone's attention, so you kind of know what's on the horizon as far as from FSIS's point of view. And so between that and the cell cultured meat, I think those are kind of the biggest things that I can think of from our perspective coming out in the future.
Great. Very good. Well, thank you so much. I really do appreciate your time. So yes, our next presentation is going to be on some of that research that was just discussed by Dr. Sara Gragg. She's an associate professor in animal science and industry here at Kansas State. And she'll be discussing validation of commercial antimicrobial intervention technologies to control salmonella and Shiga toxin producing Escherichia coli on market hog carcasses and pork products. Kind of a mouthful of a title, but I think that you'll find some of the work that she has been doing with her colleagues in the meat science department here at K-State is quite interesting and very relevant to what was just discussed. So Sara, you want to take it from here?
Yes. Thank you, Dr. Olivnik, for the introduction and for the opportunity to be here today. So as you said, we're here to talk about our research on market hog carcasses and pork products, looking at interventions to control salmonella and STEC. And I want to begin by saying that I am just one person representing a very large team of experts that has been working on this in our Animal Sciences and Industry Department and the Food Science Institute. And I'll actually be introducing some different people in a couple of slides to recognize their efforts as well. So let's go ahead and begin. We're going to start with a little bit of background and rationale. So we've already heard Melanie talk a little bit about salmonella and concerns with pork and hogs. So we know that salmonella is associated with hogs and pork. We know it's in their intestinal tracts. We know it can be found in the lymph nodes as well. We know that hogs can also harbor STEC. At this point, we feel as though there's less research on STEC than we have on salmonella in terms of hogs and pork products. And so the second part of this talk will be presenting the STEC work and really some of the first work on STEC in terms of interventions with hogs and pork products. We know that pathogens may contaminate carcasses, and that can lead to subsequent subprimal and trim contamination. So we need to really consider how we can counteract these pathogens from the carcass level all the way through to the trim components as well. So we're going to talk a little bit about what that process looks like in terms of application of interventions throughout the entire process. It's important to point out that when we're talking about market hog carcasses, they do go through a process known as scalding, which I will mention here in a little bit. We know that scalding does reduce pathogen contamination, but there is opportunity for recontamination to occur downstream throughout the harvest process. And so we really need to think about what we can do to intervene even after the scalding process, which could be things like a carcass wash, for example. So all of this means that we need validated antimicrobial interventions for the pork industry. And we really need to consider the efficacy of different interventions at the carcass level, as well as at the subprimal level, as well as trim. So we actually have quite a bit to go through today. Our team's been very busy working on this research. So I'm going to go through quite a bit of information, and I'm going to do it a little bit quickly, but of course we'll have time for questions at the end as well. So in terms of everything that we've been doing for Salmonella and STEC, we have basically two overarching research objectives. And the first is to consider how effective different antimicrobial interventions are at reducing these pathogens. And then we also are interested in what are these interventions doing to product quality. So let's start with our first objective, and that was to look at Salmonella and STEC reductions on the carcass surface. And we're talking about the outside skin-on surface, because of course when market hogs are harvested, they actually go into the cooler with their skin on. And so that's a little bit of a different surface than the interior part of the carcass, which we are considering the inside cavity for the purposes of this research. And that is primarily lean and adipose tissue on the inside there. So very different surfaces that we are trying to inoculate and treat in our research. In terms of our Salmonella research, we were also looking at subprimals and trim. And in terms of subprimals, we were looking at Salmonella reductions as well as lactic acid bacteria. Now, subprimals are typically vacuum packaged as they go out from the processing facility. And so that creates an anaerobic environment that could lead to lactic acid bacteria in terms of spoilage and issues with the product throughout shelf life. We also looked at trim in terms of Salmonella reductions as well as aerobic plate counts. Now, trim is oftentimes stored and packaged aerobically, and so that's why looking at aerobic plate counts as a sign of spoilage issues was appropriate for those samples as well. I also mentioned an interest on product quality. So we looked at carcass and subprimal color, as well as the oxidation of trim throughout the studies as well. Now, as I mentioned, it takes a very large research team to pull off this type of research. And we have many, many people that we need to recognize and highlight. So I want to take a minute here and point out that we, first of all, conducted this research here at the Biosecurity Research Institute, which we will refer to as the BRI throughout this presentation. And the BRI has a large staff of engineers and scientists that help pull off large-scale projects like these. And so over here on the left-hand side, we have a whole list of individuals who make projects like this possible at the BRI. And so I want to take a moment to thank them for all of their efforts in support of our research. Now I want to also draw your attention to the picture over here on the right. We have Katia Pozuelo on the left and Daniel Vega on the right. These are our two students that were primarily leading this effort. This was actually Katia's master's thesis work, and she was heavily mentored by Daniel throughout the process. So I want to acknowledge their efforts and give them a special thanks today as well. We also had other students working with us at the BRI, Josh Mahar, Kellen Habib, and we also had several of Dr. Trinetta's students working with us in the laboratories at Call Hall. And I also want to point out that at the BRI, we required the meat lab staff and personnel support to help us process the hogs and take our hogs to the carcass and the trim level as well. So that would be our meat lab manager, John Wolf, and PhD student in the Department of Animal Sciences and Industry, Francisco Nahar. So special thanks to them for making this project possible as well. So moving along, I want to also recognize our other faculty colleagues that were heavily involved in this project. Dr. Randy Phoebus, who is also a part of this workshop today, Dr. Valentina Trinetta, part of our food safety team, and then Dr. Travis O'Quinn, who is a part of the meat science team in our department as well. So all of these individuals really worked together to make this project happen, all the way from conception and writing the grants to, of course, carrying out the project, analyzing the data, and we're currently in the write-up phases of both of these studies as well. And I really want to give a special shout out to Dr. Randy Phoebus, and I'll comment more on his role here in a little bit, but he was the lead on the first study, the salmonella study, and put a lot of effort into getting all of our intervention technologies worked out with our contacts in the industry, worked countless hours at the BRI with the BRI staff and personnel to upgrade all of our pumps and nozzles and the chab cabinet, make sure that we could really simulate commercial standards. So special thanks to Dr. Phoebus, the BRI crew, as well as our industry contacts, which I'll mention here in a minute. And then we also had some other students who worked many hours, both on the prep side for making media and everything, all the way through keeping the lab maintained, which is Brock and Brittany and Kellen and Erica. And then we also had Dr. Kang, who is our statistician colleague on these studies. I also want to acknowledge that the Foundation for Meat and Poultry Research and Education was the funding agency for all of the salmonella work that I'll be presenting today. And then the pork checkoff through the National Pork Board was also responsible for all of the STEC funding. So a special thanks for the funding support that make these projects possible. So again, very special thanks to all of these individuals and anybody else that I may have inadvertently left off. So let's take a little bit of time here to recognize our partners. And like I said, Dr. Phoebus is also on the workshop line today. He's the one who really worked with all of these individuals at these companies. He really led the effort to get all of the different donations of product. We also had different resources in terms of pumps and feeders from like Passport, for example. We have the Zoetis chamber that I'll show you on the fabrication side that we were using for our subprimals. And then Corbion and Burco provided different antimicrobial interventions, as well as Z Corporation as well. So very, very special thanks to all of those who make this research possible in the industry. And then we are also very lucky here at K-State to have a very strong relationship with pork industry partners, as well as beef industry partners and others who really support from an advisory perspective as well. So we had many conversations with pork industry advisors to make sure we were effectively simulating commercial parameters and the different types of processing and fabrication activities that you would see in the industry. And we are very fortunate here at K-State to have the BRI, where we can simulate these things on a commercial level with a commercial scale harvest floor, chad cabinet, and fabrication side of the process as well. So with that, we're actually going to go ahead and move into the salmonella aspect of the research. Again, this was funded by the Foundation for Meat and Poultry Research and Education, which I may refer to as FMPRE for short. Our project goals for this objective was to really look at commonly used USDA approved antimicrobial interventions. We wanted to know their efficacy at reducing pathogens and spoilage microorganisms on the market hog carcass, as well as at the subprimal and trim level. And as I mentioned, Dr. Phoebus put a lot of effort into working with our spray cabinets and our wash cabinets and with our manufacturer contacts to make sure we were simulating manufacturer specifications. And that would be in terms of volume and pressure and so forth. And I'll give those parameters in a minute in detail. And again, we used the meat harvest and processing facility at the BRI, and we were carrying out the research under biological safety level 3 conditions. In terms of bacterial reductions, I mentioned we were looking at salmonella, lactic acid bacteria, as well as APCs. And we looked again at quality, particularly on the loin. We looked at color, and we also looked at lipid oxidation from the loin and trim as well. We looked at a variety of antimicrobial interventions, and we focused on high volume as well as low volume. So one of the benefits of the chad cabinet at the BRI and as well as in the industry is that there are different phases, and the phases can provide different volumes. So we were very fortunate to have the opportunity to look at different interventions in a high volume capacity and then others in a low volume capacity. And again, this was designed to mimic what might be done in industry. So on the high volume side, we had an ambient water control. We also had a 160 Fahrenheit water wash. We used Microtox 5, which was provided by ZE Corporation, which is a parasitic acid product at 400 parts per million. We also used Porsabrome, which was provided by Passport, which was all referred to as DBDMH at 400 parts per million. And then we also looked at Porsabrome at 600 parts per million. On the low volume side, we were looking at parasitic acid 400 parts per million as well. We also included Purac from Corbion, which is a lactic acid product that was heated to 130 Fahrenheit. And we used this at 3% on carcasses and 2% on subprimals and trim. We also looked at acidified parasitic acid at 400 parts per million, which was acidified using Titan, which is provided by Zoetis, to a pH of 1.2 for carcasses and a pH of 1.4 for subprimals and trim. Now this slide gives a breakdown of the entire process that we went through for our salmonella objectives. It's very busy. You can tell it was a very large project. It was very, very involved. And each color is really intended to break out the different aspects of the study. And so on the left here, we have the blue column, which would have been our day 0 and our harvest day moving through the intervention stage. Then we moved it into fabrication the next day, pulling the carcasses out of the cooler and broke them down. And then the next day, we also used the trim for a trim study. And we're going to go through each of these aspects in more detail. So let's actually begin here with the day 0 activity. So first we started out with 4 market hogs per replication. And we actually harvested them using USDA approved procedures. As I mentioned, this took place at the BRI with our meat laboratory personnel who are very, very knowledgeable and experienced in these procedures. And so we were very fortunate to have them join us at the BRI. And so what I really want to move through here is, I mentioned the scalding, which that was around 60 Fahrenheit for 3 minutes. The carcasses they would then be de-haired and polished. So that was kind of a singeing procedure that would help us to remove any last hair from the carcass. And they then went through an ambient water wash, basically to just remove any of the singed hair that might still be remaining. They were then split and eviscerated, which I want to go ahead and acknowledge John Wolf here. You can see him working in the BRI. We then paint inoculated and allowed for attachment. And here we have Dr. Randy Phoebus performing the paint inoculation on one of our carcass sides. And paint inoculation is literally what it sounds like and looks like. It is a paint roller. And we followed a very specific procedure of how many times we paint inoculated each side. And we actually used a cocktail of salmonella that we got in terms of the isolates we got from Dr. Brian Lubers at the College of Veterinary Medicine. He provided those isolates to us that he had actually isolated from swine. So here you see Dr. Phoebus doing the paint inoculation. We then would allow a period of time for attachment. And after the attachment period, you can see on the right, there are some core samples taken out of the skin surface. And we took those core samples after attachment time, but prior to intervention. And that was considered our time zero in terms of what we would compare all of our salmonella reductions back to. And we took cores, as you can see, from the top, middle, and bottom of the carcass. And at every sampling time point, we took 2 cores from each region of the carcass. So after we collected our initial core samples, they were put through, or the carcasses were put through the Chad cabinet. And you can see here in the picture on the left, you can see the Chad cabinet in the background and all of the different pumps and feeder and tank systems that we used throughout this study. And like I said, we are very fortunate to have a lot of industry support, as well as support from the College of Ag and the Department of Animal Sciences and Industry, because for us to have all of this equipment required a lot of either donation or loaning or purchasing of the different tanks and pumps that you see there. So I want to make sure I point out and acknowledge our appreciation for all of the individuals who made this possible. So we then put the carcass through the Chad cabinet, and we then pulled cores again off of them as a post-treatment sampling time point as well. Now, after we pulled those cores, which you can see here on the picture on the right, we then allowed the carcass to go into the cooler to chill overnight. And this was dry chilling, I should also mention. So here's actually a video of our carcass wash cabinet. And you can see here on the right, we are providing all of the different parameters that we followed for the high volume as well as the low volume washes. And this video here is actually showing a high volume carcass wash. You can see how the tremendous amount of water and pressure actually caused the carcass to swing in that part of the Chad cabinet. After the carcasses cooled in the cooler overnight, it was time to fabricate them. So as I mentioned, John Wolf and Francisco from the meat lab returned back to the BRI with us, and they helped fabricate the carcasses to obtain our subprimals and trim. So as we were breaking down the carcasses, we were really focusing on obtaining the butt, the loin, and the trim for future testing. So we first started out with getting the butt, and we re-inoculated the butt with salmonella. And you can see here in the picture on the left, the way we did that was putting the butt in a bin and in a biohazard bag. And we had a spray bottle with our salmonella inoculum that we had calibrated to spray a certain amount, given a number of sprays, onto that butt surface. And we then, of course, allowed a period of time for attachment for the butt as well. After we allowed for that attachment time, you can see on the right, we're sampling cores from that butt sample to get a pre-treatment sample, much like we were obtaining on the carcasses. So this sample that was taken post-inoculation but prior to treatment would, of course, then be our reference point for comparing our salmonella reductions achieved by our antimicrobial interventions. Now, before we move on, I want to point out that all of these carcasses, of course, would have been treated with some intervention at the carcass level as well. So we were very careful to match up those carcasses that had received an intervention to the same intervention on the subprimal side as well. So in terms of applying subprimal interventions, we have a subprimal wash cabinet here. You can kind of see on the left. Again, Dr. Phoebus and Daniel Vega spent a lot of time dialing in this pump here that you can see to make sure that we could mimic commercial scale procedures and wash parameters. After we sprayed our interventions, you can see here on the right, we were also collecting another sample that we would then use as our post-treatment sample to determine salmonella and spoilage microorganism reductions. I should also point out that we used the loin from all of the carcasses, but we did not re-inoculate the loin. So we considered the loin to be non-inoculated. And that non-inoculated loin was, again, used for some different tests throughout the study that we'll talk about. After treatment, we then needed to provide subsamples from the butt as well as the loin for us to store throughout shelf life and sample after day 0. And the butt and the loin had different sampling periods, which I'll talk about here in a little bit. But both of them were cut into their individual portions and then vacuum packaged so that we could then conduct more tests throughout shelf life. So here's another video using our subprimal spray cabinet. Yes, no worries. So what I was mentioning was that in that subprimal spray cabinet, the antimicrobial intervention is really hitting the subprimal from all directions. And it was coming along on the belt. We captured it at the end as it was coming out the back end. And then we were able to allow it to drip for a short period of time before collecting our post-treatment sample cores from the butt and the loin as well. So the next aspect of our study was to use the trim that we collected from the fabrication portion of the study and have two portions of trim. Actually, we had a portion of trim that we re-inoculated with salmonella and a portion that we did not re-inoculate, just to look at other spoilage microorganisms throughout shelf life. So we re-inoculated the trim in much the same way that we did the butt. You can see the same picture below, actually highlighting the same spraying technique. And then you can see on the right a picture of what our trim product actually looked like. And we would, of course, re-inoculate the trim, allow a little bit of attachment time, much like we did with the other products. And then we would sample prior to treatment to get a pre-treatment sampling point. We then used the same treatment approach that we did for the subprimals, meaning we matched the same trim product to the carcass that it would have originated from in order to match the same interventions. And we actually tumbled our trim. You can see here in the tumbler in the picture on the left, we used 120 mils of intervention that we actually sprayed using a garden sprayer. So we would tumble, stop the tumbler, spray intervention, tumble for another minute or so, stop, spray more intervention until we had a total of 120 mils sprayed and a total tumble time of 3 minutes. So after the tumble time, there was a little bit of a drip period as well. And then the post-treatment samples would have been collected. And the way that we sampled these was to basically pull representative pieces from throughout the entire batch, similar to N60 testing that is used in the industry. And then our trim would have been stored under aerobic conditions. So next I want to talk a little bit about the sampling process as well as the different types of analyses that we conducted on all of the samples that were collected. So over here on the left, you can see we have the carcass hanging again. We, as I mentioned, collected samples pre-treatment, post-treatment, as well as post-chill. So as the carcasses were coming out of the cooler, but prior to fabrication, we collected more core samples. And again, from the top, middle, and bottom of the skin-on surface, as well as that interior cavity. And these core samples ended up being 42.1 centimeters squared and were collected as tissue excision samples. So you can see actually in the picture the indentations where we removed those core samples. The cores were immediately put into day-Angley neutralizing buffer, which is important because that neutralizing buffer stops the antimicrobial activity so that our samples don't continue to basically kill salmonella or spoilage microorganisms post-sampling. We want that antimicrobial activity to stop upon sampling, which is why that day-Angley buffer is so important. We then took those samples back to the laboratory here in Call Hall and analyzed them for salmonella. Subprimal sampling was conducted in much the same way. So we would have taken core samples at pre-treatment, post-treatment, and then throughout storage as well. We did collect smaller core samples for the subprimals because they were smaller in size than the carcass. But it did end up being 38.4 centimeters squared. And again, that same tissue excision sampling method that you can see in the picture was followed for both the butt and the loin. Now, it is important to point out that we felt like the loin and the butt, of course, had different composition in terms of lean versus fat on one side versus the other. So in order to get a representative sample, at every sampling point, we collected 2 cores from the lean side as well as 2 cores from the fat side. And then much like the carcass samples, we also used day-Angley neutralizing buffer for the subprimals as well. As I mentioned, the butt was re-inoculated with salmonella. And so at sampling points, we were analyzing for salmonella in terms of CFU per centimeter squared on days 0, 7, and 14. And we were actually looking at salmonella reductions. The loin, as I mentioned, was not re-inoculated with salmonella. But because this product would be vacuum package stored, there is a risk for spoilage to occur from lactic acid bacteria. So we actually used the 3M aerobic plate count Petri film with MRS broth as a diluent to basically create a medium that allows for LAB to grow on Petri film, because 3M actually doesn't have a lactic acid bacteria Petri film that was going to work for our purposes in this study. So this was the way that we decided to use Petri film to look for LAB, and this is actually a published method. And we were conducting these analyses on days 0, 7, 14, 21, as well as 28. We also used the loin to look at lipid oxidation, and using a method called thiobarbituric acid, or what we also refer to as T-bars for short. And you can see here in the picture the different test tubes that were used as part of the T-bars analysis. And this would have been something that Dr. O'Quinn would have mentored Katya and Daniel through as part of our study. And then likewise, Dr. O'Quinn also mentored our team through the color process using a colorimeter using the loin. So we would have collected color at various stages throughout the treatment process. Trim, as I mentioned, we were using somewhat of an N60 testing idea in terms of collecting representative pieces that were a part of the trim batch that you can see here on the right. And we conducted our micro testing on day 0, 2, and 4. Now, because this was re-inoculated, we were able to look for salmonella and to calculate reductions achieved as a result of our antimicrobial interventions. But I did say that we also had a portion of trim that we did not re-inoculate, but that was treated. And we used that trim to look at aerobic plate count populations. We also used the non-inoculated but treated trim to look at lipid oxidation using T-bars as well. Let's move along now into the reductions and the results portion of this study. So as I mentioned, we used the different sampling points to look at salmonella reductions. And we again would have compared all of our reductions back to that time 0 time point. So that post-inoculation but pre-treatment sampling point. And we looked at cores from the carcass exterior, so that skin on surface, which is what is represented here. And what I really want to point out is that you can see that we achieved anywhere from about 1.5 to 3 log reductions with all of our intervention technologies, whether that be low volume or high volume. And we have our salmonella reductions over on the left-hand side and our treatments along the bottom. And you can see that our first 3 treatments, so the high volume PAA, the high volume DBDMH 600, and the high volume hot water, they were all the largest in terms of reductions achieved for salmonella in this study. And then, as I mentioned, the other technologies also achieved anywhere from 1.5 to approximately 2.5 log reductions. So what this really tells us is that our industry partners who might be looking to implement a antimicrobial intervention technology in their facility really have a variety of choice in terms of what would work for their facility that also is validated to reduce salmonella by at least 1.5 logs and maybe up to 3 logs. And so that was really what we were going for in our study, was to provide a bit of a toolbox of validated options for processors to choose. So for example, some facilities might have capacity to do high volume. Some facilities might have a low volume capacity. And so that was why it was so important for us to look at high volume and low volume options for the industry. When we look at the carcass interior, we definitely did not see as large of reductions. And so as you can see here, the largest reduction achieved was by the high volume paracetic acid, which was up around 1.7 logs. And then that varied all the way down to our lowest at the ambient water that was applied at a high volume. You can see only a 0.1 log reduction was achieved there. So in general, what we really noticed in this study was that it was harder to achieve a salmonella reduction on the carcass interior. Again, that could be for a variety of different reasons, but we know that that skin on surface versus that interior lean and adipose tissue surface, we know those are very different surfaces. We also have to acknowledge that that interior carcass surface is somewhat shielded by just the anatomy of the carcass, which could also be impacting the ability of our interventions to have good coverage throughout that interior cavity. I also want to point out that in terms of quality subprimals and trim, we did not see data that were significant, particularly in terms of treatment. So what that really tells us is that our antimicrobial intervention technologies were not as effective at reducing salmonella populations or LAB and APC on subprimals and trim. So very different in terms of what we saw on the carcass surface. We also want to point out that our antimicrobial interventions did not cause lipid oxidation and did not negatively impact color. In conclusion, when we look at the salmonella carcass data, I'll begin by discussing the exterior skin on surface. The highest log reduction that we observed was around 3 logs, and that was for the high volume chemical and hot water wash. The low volume sprays achieved around a 2 to 2.5 log reduction in salmonella. In terms of the interior lean surface, there was that 1 to 1.5 log reduction that we were just discussing, with the largest reductions achieved by the high volume PAA, the low volume lactic acid, and the high volume DBDMH 600. Now the other treatments achieved a 0.5 to a 1 log reduction. And I also want to point out that air chilling contributed an additional 0.5 log to 1 log reduction in some cases. So we do know that sometimes salmonella will continue to decline throughout that chilling period. So moving along, I have just a little bit of time left. I want to touch base on our S-Tech work, which was done on the pork carcass surface. And again, this was funded by the Pork Checkoff. Now in this study, our project goals were the same as previously mentioned in terms of looking at commonly used USDA approved antimicrobial interventions. And we were specifically looking at the market hog carcass surface. In this study, we actually stopped at the carcass level. We did not go through the subprimal and trim components that we did for salmonella. We were looking, that is a typo, my apologies. We were actually looking at S-Tech reductions and then impact on carcass color in terms of lean, adipose, and skin. We looked at the same antimicrobial interventions as previously mentioned. And we also followed the same procedure in terms of using USDA approved harvest procedures from our trained and experienced meat lab personnel. We, of course, followed the same splitting and evisceration, as well as paint inoculation and attachment procedures. So in this case, we would have been using a cocktail of S-Tech. So we would have used a strain representing E. coli 0157H7, as well as a strain from all of the big 6 adulterant serogroups as part of our cocktail. So we would have had all adulterants represented in our cocktail for this study. And followed again a very similar, or actually the exact procedure for inoculation, as well as pulling samples both pre-treatment, post-treatment, and post-chill. The antimicrobial interventions were applied in the same way, going through the CHAD cabinet using the same infrastructure and setup as well. and then followed by the same cooler procedure, which would have been overnight stored at basic or without any, it was air chilling. As I mentioned, carcass color was collected in this study, and we were looking at a variety of locations trying to determine if any of our antimicrobial interventions impacted color, whether it be on the lean tissue, the adipose tissue, or the skin on surface as well. And again, Dr. O'Quinn would have been our faculty colleague helping us with this aspect of the study. We would have looked at carcass color at pre-treatment, post-treatment, as well as post-chill. Here is a table summarizing our data. It's a little bit busy, so I'm just going to really point out to you some of the main things that I think are of interest here. So we can see that our top 3 treatments here achieved anywhere from 2.8 to 3.3 logs, and those were all statistically the same to one another, and that would have been post-treatment. However, all treatments post-treatment achieved anywhere from 0.7 up to 3.3. So we had quite a variety in terms of efficacy at reducing STEC on the carcass surface. And this was the exterior skin on surface, I should also mention. In terms of post-chilling, those same 3 interventions achieved a 3.8, 3.2, and 3.4 log reduction, as you can see here on the right-hand side. And as I mentioned previously with our salmonella work, we also observed with STEC that just the chilling procedure can achieve an additional 1 log reduction in the case of our hot water treatment applied at a high temperature. I also want to point out the same thing occurred here with our lactic acid treatment. From post-treatment to post-chilling, we went from a 0.7 log reduction in STEC to 1.6. When we look at the internal body cavity, kind of like salmonella, we see the same trend with STEC in terms of we didn't achieve as large of reductions. We are top 3 performing interventions achieved anywhere from 1 to 2.1 logs. And then when we moved to post-chilling, we had anywhere from 0.9 to 2.2 in that 1 to 2 log reduction range as well. And again, with lactic acid on the inside cavity, we saw an additional reduction during chilling from 0.6 to 1.1 logs. In this study, we also observed a significant effect in terms of location on the carcass. So if you recall, I said that we sampled at the top, middle, and bottom. Well, in this study, we achieved the largest reduction at the bottom of the carcass, which was actually statistically the same as the top of the carcass. And then the lowest reduction was actually achieved in the middle of the carcass. And there's a variety of reasons that that could be. One, it could be that just the middle, for some reason, didn't receive as much intervention. Also the bottom, as the intervention dripped off the carcass, the bottom of the carcass may have actually received more intervention contact time ultimately as it dripped down. Just one hypothesis as to why we may have seen the data turn out this way. So in conclusion, with our STEC data on the carcass surface, on the exterior skin on surface, we achieved the same approximately 3 log reduction for STEC that we did for salmonella. Again, using high volume chemical and hot water washes. The low volume sprays achieved anywhere from 0.7 to 2.4 logs in terms of reducing STEC. And on the interior surface, a 1 to 2.1 log reduction in STEC was observed using the high volume PAA, low volume PAA, and the high volume DBDMH 600. And other treatments achieved 0.2 to 0.8 log reductions. And we saw, again, the same trend with STEC that we did with salmonella, in that air chilling contributes an additional 0.5 to 1 log reduction in some instances. I mentioned carcass color was also collected, and our color data were actually not significant. So what that tells us is that our antimicrobial interventions did not negatively impact color on any of the tissues. So that included the skin, the lean tissue, as well as the adipose tissue. Now I have a little bit of time left. I want to take just a few minutes here to compare our salmonella and STEC data at the carcass level. This is a little bit busy of a figure. I'll kind of quickly point out a couple of things here. We have our log 10 CFU reductions on the left, and then we have our treatment along the bottom. Salmonella is represented in the dark bars, and STEC is represented in the lighter bars. And these are reductions achieved on the exterior surface. So what I want to point out is that those same three interventions that we were highlighting for both pathogens really achieved pretty similar reductions for both pathogens. In fact, most all of these interventions achieved pretty similar reductions. Probably the biggest exception would be the low volume lactic acid spray that you can see here towards the right of the graph. That seemed to be more effective against salmonella than STEC on that market hog exterior surface. When we look at the interior surface, we have a little bit of variability there in terms of efficacy, which, you know, we have been talking throughout this presentation that the antimicrobial interventions did not achieve as large of reductions on that interior surface. And we also see a little bit of variability in terms of efficacy against salmonella versus STEC. So looking at the interior surface in future research is probably an important research opportunity to try to optimize some of these interventions and understand what we can do to improve reductions on that surface. So some overall conclusions of our work. We saw similar reductions in salmonella and STEC. Approximately that 3 log reduction was achieved on the carcass exterior for the same high volume chemical and hot water washes. And it is important to note, once again, that over 1 log was achieved for all interventions. And so that's important for recognizing that hog producers and market hog processors really would like to have choice in terms of what's right for their operation. And so it's really important for us to point out that these interventions provided some level of efficacy on the carcass surface, which is definitely important. There was indeed variable efficacy on that internal cavity surface, the lean tissue versus skin tissue. As we mentioned, those are very different matrices. And so attachment and ability to reduce pathogens would be quite different just by nature of the surface. And then again, as I mentioned already, that anatomical difference in terms of that inside cavity may also be contributing, and that contact angle of the intervention may be difficult to get good coverage on that inside surface. So with that, I'll go ahead and stop here and invite any questions that you might have. And thank you very much for attending today. And thank you again for the opportunity to be here.
Thank you so much, Sara. My goodness, that was a lot of work that was done in those activities, and I can see that there's some really useful information that was produced from it. We do have one question here that came in over the YouTube. Says, can you discuss the importance of recognizing a 2 to 3 log reduction at the carcass level with a 5 log inoculation level versus achieving a 1 log subprimal reduction from a 3 log inoculation level?
Sure. So it is important to note that us putting in or achieving a 3 log reduction is pretty impactful, considering we did aim to inoculate around 5 logs. So the reason we inoculate at such a high level, either on the carcass surface or the subprimal and trim, is so that we can actually observe the reduction that we achieve. So if we only inoculated in this case with 2 or 3 logs, we wouldn't necessarily know how effective our interventions were, because we would have achieved that 2 or 3 log reduction and wondered, could we have achieved 4? Could we have achieved 5? So by inoculating at the 5 log, we were able to see the full 3 log reduction that was achieved. And so when we think about the fact that we artificially put 5 logs there versus what would maybe naturally be found in terms of what carcass contamination would be, that 3 log reduction is very significant, because it would be, I would say, on the market hog carcass surface, to see 3 logs of contamination would be very high. And so to know that we can achieve a 3 log reduction on that carcass surface should be very effective in terms of what natural contamination levels would be, and to control and reduce those to an acceptable level on that carcass surface. So that would be the part 1 of the question. And then in terms of the subprimal and trim, as I mentioned, we achieved kind of up to a 1 log reduction there, but and that is impactful as well, because we, in general, in terms of subprimal and trim, whether it be beef or pork, it's oftentimes very hard to achieve reductions on those chilled surfaces. And so for us to achieve 1 log is actually pretty meaningful. Now, we may not have necessarily seen as many significant reductions, but that does tell us that some of these interventions are showing some efficacy at the subprimal and trim level, that maybe with some more effort we could optimize them and try to improve those reductions as well. So that's why even a 1 log reduction is still meaningful at this point.
Right. I've got a question on methodology of getting this information out. So again, you had a huge amount of research that was carried out, and a lot of information that showed improvements in reductions of various microorganisms. What's the process of getting that kind of information out to the actual producer? And has this been taken up in production facilities, and how does that system work?
Yeah, so actually Dr. Phoebus has already given some presentations that the industry would have been able to participate in and to be a part of. And so this information is already being distributed, and particularly on the salmonella side. So the salmonella research completed probably last fall, about a year ago. We've been analyzing data and writing it up since then, and Dr. Phoebus has started disseminating that information. Now, the STech information is actually newer. We actually just finished that work up here not too long ago. And so that work is now just starting to get written up and get ready for dissemination. So we will continue to give these presentations to share this information with industry. And our next goal is also to come up with some peer-reviewed publications very soon to go out for the industry to use as a scientific peer-reviewed piece of information for them to use and make decisions for their process.
I might send that exact question to Dr. Abley. How is that information that's developed at a university setting like this utilized within your area?
Great question. So once we become aware of information like that that's out there, if establishments ask us questions specific to different interventions, and they don't know where to find scientific support, or they're not sure it exists, if we know that there's some out there, we will point them directly to that information. And in addition, we will include this information in our guidelines. And we're currently working on updating our draft salmonella guideline for market hogs right now. So yes, this study would be very great once it comes out. If you could send a link, we'll be able to add that to the guideline that's currently in process right now.
Maureen, this is Randy Phoebus. Can I just have a little bit of input here?
Sure.
Both of these studies that Dr. Gragg just presented are funded directly from the industry trade organization itself. So the Pork Board and the National Cattlemen's Beef Association, American Meat Institute. And because they fund it, they expect us very much to put interim reports and a really detailed final report forward. And so we have done that, and they have actually disseminated it and made it available to all of the industry, their members, which is most of the industry. And so those reports, particularly the final report, are already out there in circulating, and you know, they're not truly peer reviewed, but they're kind of pseudo peer reviewed with all the statistical analyses and things like that. So hopefully within the next couple of months, we will have a true journal article out of each one of these. But right now, those final reports to the funding agencies are out there and are very valuable to the companies making decisions.
Perfect. That's an important bit of information from the research side as well as the user side. So thank you so much. And thank you, Dr. Gragg, for this presentation. It was a great overview, and I think it goes to show some of the high level of research that's being done in the area of meat safety here at K-State.
Thank you for the opportunity.
Okay, now next up we're shifting gears just a bit. We've got Dr. Gordon Smith, who is the director of the International Grains Program Institute, and he's also the head of the Department of Grain Science and Industry here at K-State. And what we've asked Dr. Smith to talk about was the intersection of industry and academia in solving food safety challenges. And he'll give you a little bit of background on himself, and you'll understand why somebody, the head of the Grain Science Department, is talking about meat safety. But we look forward to this discussion. And Gordon, do we have you?
Sorry about that.
Thank you.
All right, so good morning, everybody. As Maureen said, I'm a diversion, and so Dr. Abley and Dr. Gragg are hard acts to follow, and they've given me a fair amount of PTSD, which we're going to talk about in just a second. So, you know, this, see if I can advance it. This is me in a few pictures. And so I am the head of the Grain Science Department, proud to be so. We have great faculty in a very unique position. We're the only department of its kind in the United States, and with a small number of faculty, have quite the research and extension and teaching impact. And so again, a department very much tied to the industry, but I'm not a grain scientist. I'm a food scientist, and before that I was a chemist. So I have an undergraduate degree in chemistry and biology, a master's in organic chemistry, and a PhD in food science. And my food science degree came from within the animal science department, and specifically the meat science section at Texas A&M. So I was trained as a meats guy, and because of that, I was hired by Sara Lee, and at the time Sara Lee was run in divisions, 36,000 employees all over the world. And I was hired by Jimmy Dean, and I was hired by Jimmy, who was still alive at the time. So I had a chance to work for the entrepreneur in a big company. I was also jointly hired by Sara Lee, kind of senior administration. And so because of that, that's how we got here. So I'll just read the right part of this, you know. So in addition to killing at least 15 people, miscarriages, sickening 80 in a number of states, Sara Lee in December of 1998 announced the recall of 50 million pounds of meat, which would later be about 35 million pounds. And it wasn't December, actually, it was December 21st. And I remember that because on December 20th, as I was in shorts and t-shirt, loading up 2 kids in the back of the car, getting ready to go home for Christmas, I got a phone call from my boss's 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 boss, George Bryan, who ran all of Sara Lee Meat's operation for his brother, who's the CEO of the corporation. And again, Sara Lee is a big business, you know, at the time. And so multi-billions of dollars. George Bryan's admin assistant called me and said, Mr. Bryan wants to see you. And I go, okay. And she goes, he wants to see you now. And we were in a suit and tie organization. So I said, I'll have to change clothes. And she goes, he wants to see you now. Come like you are. And I go, I'm wearing a shirt. I'm wearing shorts and a t-shirt. And she goes, you need to come in. So when we tell this story to students, You know, I expected I would be fired, right? But when I walked into Mr. Bryan's office, he was chewing on a cigar back in the old days in a huge office with a corporate lawyer, and he handed me an envelope, a letter, and the letter was from USDA. And it said that they were going to give us until tomorrow to make a decision about what we do with the operations at our Bilmar plant in Michigan. And to be clear, I didn't work for Bilmar. I didn't work for the plant nor for the division. But George had helped to hire me. I was one of 2 PhDs for the corporation, and he thought that I should know what to do, which I find really funny, seeing how I was a really young scientist at the time. And that became a 30-month journey into food safety. Dr. Phoebus and I, who met long ago, talk about this all the time. I was a chemist thrown into a microbiological world when food microbiologists were much less common than they are now. And we lived with my job essentially was the corporation's response to safety, not the plant response, not what was going on at Bilmar, but what Sara Lee was going to do in the face of a recall that cost us about $400 million. And that was the profitability for more than a year of the corporation. So I learned this in combat. It's near and dear to my heart. It's funny how many memories even the lectures that we heard this morning do. And so I spent my life until 6 years ago in the industry. I was always interested in academia. My father's a college professor, and that put me in a variety of roles where I was responsible for budgets and managing projects between the companies I worked for and academics. And I guess at the high water mark, we had a budget of about $2 million a year that was distributed among a number of projects, but it was more than a trivial operation. So we're going to spend the next few minutes talking about, you know, the how and the why that academics and industry people can work together for the good of both parties, right? And so just 2 or 3 slides really quickly to kind of level set everyone to begin to think about R&D, research and development from the industry's perspective. Is the things that you do depend on the size of the business. It could be formulating a new product on the bench today that we're going to launch tomorrow. It could be quite complicated research, but in general, the larger the organization, the more complicated the task that organization performs. Again, with size comes, you know, specified expertise and different levels of education. And also with size comes hierarchy. And so in a small organization, it could be that you report to the president. The R&D person might very well report to the person who's making decisions. In a larger organization, there might be a number of layers. Most of the companies that have substantial research with academia are medium to large. They're not necessarily large, but not in general small. Small organizations might work through the associations, as Randy has just kind of talked about. And then the functional areas for these people are a variety. Most of these you recognize, certainly food safety, but technical service means those are the plumbers. Those are the guys who are called when things are happening generally in operations, but sometimes in distribution that require a technical solution that's beyond the ability of the operations people or the distribution people who are directly in charge. So they call the Maytag repair guy. That was how I started my career. And you would walk into a plant and have to figure out what was going on and why. And that could be beyond product development. It could be just about pure operations, getting things out the door, or it could be about safety or regulatory compliance. So this is just an example of what an organization might look like. This is from when I left ConAgra 6 years ago, but these are the pieces. And so these aren't just food scientists. These are lawyers and sensory experts and a variety of other people, nutritionists and finance people and marketing people, all that work within the R&D organization. So this isn't the people we work with. These are the people that were employed by, that were on the payroll of research quality and innovation. So again, quite a number of different skill sets as you think about who you might partner with. This is consumer-driven product development. Again, The importance to meat safety, to food safety is, you could argue it takes a formulation for us to understand and make assessments on shelf life and safety, but it's the second point under the second question here, and it's critical, right? Not only do you have to have products that are safety, as Dr. Abley said, that meet the regulatory requirements of USDA in a meat product, but it's also good business. Businesses don't want to hurt consumers. They don't want to produce things that are going to cause them regulatory products. And so they consider that up front. And that's what we talk about as we talk about technology designed to meet not only regulatory expectations, but also product guideline expectations. So why do we need to collaborate? What exactly is the reason that drives industry to academia? And so again, by listening to the people, and you're going to have Dr. Phoebus after me, the bottom line is you see the kind of expertise, specific, in-depth, long-standing, very in touch with what's going on in the literature kind of expertise. Generally not found except in the largest of R&D organizations. Nestle, General Mills, others would have that kind of expertise, but it's the purview of a few. And with that expertise comes a great depth of understanding about the science and technology. And so why else? We always talk about cross-functional teams, but it is critical to this, right? Is having someone here at a university that understands where the other resources are important, and the industry oftentimes understands also that while it might have strength in one place, it doesn't have strength in other places that are required to solve the problem. And the next three are really interesting, right? Is sometimes smart business understands an external view is important. No matter how good you are, you tend to get blinded by internal realities. And so sometimes it's just good to have somebody walk in and tell you like it is. Part and parcel with that, right, is this idea of validation. That I know what I think this does, but what I really like is someone who's recognized from the outside to come in and validate what it is that I think. And through that validation process, especially if the data is good, then the validation work can stand of its own, and it can be an advocate to what the industry wants to do. Not suggesting you pay academics for answers. I'm suggesting that the answers are in line with what you need, then you can use that work and you can use the person that conducted that work to advocate. I don't think that's a problem for industry. I don't think that's a problem for academics, assuming it's data-based. Academics can bring solutions to the business. We just talked about it, is we do work in the BRI. We have smart people here that have ideas, and they can come to the business and say, we have something that we think will solve a food safety or a regulatory project. And then the asterisk at the bottom is academics can be excellent partners and not. And so we're going to spend just the next few minutes talking about that. So what makes great collaboration? That the collaboration is the best tool for the job, and that everyone recognizes that. And generally those conversations occur in industry before you go out to academics. You get buy-in from senior management, quite possibly cross-functionally senior management, that it's time to involve academics. And that is how you get to the second piece, which is the higher the support that for this collaboration is in the organization, the stronger it is. So you'd really like to have the CEO or the SVP of R&D or technology who are both aligned with the specifics of what the collaboration is. Academics help by being known outside the R&D organization. So if someone knows, Dr. Craig or Dr. Phoebus through whatever, they've seen them speak or they've seen a popular press article that they've written, that helps. And so, having these business people that somehow know academic experts is a good thing. Project goals have to be understood and committed to, and we're going to come back to that over and over and over. And then, you know, the next one may be the hardest, is people in academia. This is a very specific culture, and it's very different than the industry culture that I came from. I knew that before I came here, having lived around my father, a college professor, you know, for 18 years. And so these cultures are quite different, and working with the other side, working with industry people if you're an academic, or working with academics if you're an industry person, is a lot different than understanding those organizations. So a whole lot of people who spent time working with industry, that's different than being in the industry. And I know that on both sides, as it turns out here. And so you need the right obligations, you know, the obligations, you'd be clear, you need the right agreements in place. So what does each party bring? You know, what do you get? So what do industry people bring to collaborations with academics? Well, they bring a knowledge of the marketplace. In large company, millions of dollars are spent on understanding the products that you have, the products your competition has, the consumers, the customers, which means who you sell to in the first pass. That's Walmart and Kroger's and Dillon's and Costco and Sam's. The specific products that a company makes are things that they've been tweaking with at a variety of levels. So it could be R&D, could be operations, could be a lot, it could be QA folks, but they have been tweaking with products sometimes for decades. And so they understand, you know, this idea of what they've got and how they got there, both in the present and in the past. There's quite a bit of technical know-how. Some of it's based on data and some of it isn't. The most interesting conversations in an industry perspective are when somebody swears the situation is a certain way and a data-based person comes in and says, well, I'd like to see the data. And 5 years later, we're still waiting to see the data. Because if the data exists, no one knows where it is, or it may not exist. It may be sheer, urban legend, if you will, but both exist. There's a great deal of historical information within companies about products that isn't shared externally. And there's a huge amount of information regarding of what's been tried and what's not. It's the biggest problem with using external product development, right? Is outside the company, others don't know that you've tried 200 things and you've tried things that are in the marketplace that might have, in the marketplace now, that might have failed. I have slides of things that were discussed 30 years ago that have been in the marketplace the last 5 years. And so that is kind of, what the industry has. What's the university have? University brings people that can explain to you how things work, and business people, even technical business people, deep down inside want to understand the how. They want to understand it at a level that they can understand it at, right? So, having Dr. Phoebus and Dr. Gray go full, PhD microbiology on me as a CEO of a company is not the right approach. They need to use that expertise and explain the situation at probably a, freshman in high school sort of level, to bring who are very smart people along in scientific fields that they just haven't had the exposure at any point in their formal or informal education. We talk about collaboration. Industry looks to universities for internal experts to connect them to other internal experts. How do we get other people connected? How do we get the right people connected? Dr. Phoebus and I worked on a project for a major food company because they contacted me, but I knew that I needed more microbiological expertise than I possessed. I had some of the puzzle, but I didn't have all of it. I needed somebody else, and I brought in somebody at K-State that I knew could provide that expertise. People in the industry don't have any marketplace bias. You don't know how much things cost. You don't know what my margins are. You don't really know very much about my consumers or competition, again, to the level that the industry does. And so you're really not biased by those things, which is a good place to start. At some point, you will have to consider, you know, the operational feasibilities and the costs, but in the beginning, you want the funnel to be very wide. And you have no company bias. So you don't know what you're allowed to say and what you're not allowed to say. And you don't know when you say, you know, we really ought to fill in the blank, that you've just made the head of that department or the other department quite angry because they've been opposed to that for a decade. And certainly this idea of the outsider's voice, sometimes there's great power to the voice of the outsider. So what are common misconceptions about collaborations with industry? This is my favorite. When I came to K-State 6 years ago, what I heard from faculty and administrators alike was industry funding isn't the same as competitive funding, meaning competitive government grants. And industry funding isn't competitive. So if I compete for USDA grant or FDA grant or DOD grant, that is one thing. But if I get money from, you know, General Mills or ConAgra or PepsiCo or somebody else, that's something different. And I would tell you, there's absolutely not a fiber in my body. Industry funding is incredibly competitive. And I fought for the funding I had for academics every year. And the academic projects that competed for that pool of money were always much larger than the pool of money was. So there's quite the competition internally to get the funding, and then with the external parties to get access to what is a shrinking pool of money. Technical knowledge is valued. People said at the university, go, this stuff's important, science is important. And sometimes in industry it is. And sometimes in industry, the technical knowledge is lost. And what's important is how you turn products into profit. That's what business is. And it's not bad, but it isn't always valued the same as it would by someone who's immersed in the field. You know, the real research and development is done in academia, and what's done in industry is really just rip and apply. When I came to K-State, I had several papers that were done with academic partners, a number of them, as a matter of fact. And one of the people on the search committee here called one of my academic partners and said, did Gordon do anything to be on this paper, or did you just put his name on it out of courtesy? And when I die, I won't forget that story, because that was my research, it was my idea, it was my design, and I executed it. And I involved academics for a lot of reasons, but it was mine, right? Yet the perception was, it was probably, you know, I was probably just riding along on somebody else's research. Relationships are critical, and they're not just between the people doing the work, but also at other places within the organization. You know, again, the industry understands this. It should be clear to most academics that that isn't always true, and that academics have a great opportunity to help industry understand better. And then either party is easy to work with, not necessarily. And sometimes it's situationally dependent. So it's not just personalities, but it is timing and overriding circumstances. Certainly the COVID is one of those, but again, it requires understanding. So how about working at the interface? So I'm going to kind of close with a little bit of this is what matters? Well, deliverables matter. My entire life, when I had a project with academia, I had committed to the organization and oftentimes to the CEO that there would be information along a certain time continuum. And, you know, people understand that research doesn't always work that way, but you do owe them an update, and you do owe them understanding about when they're going to know something, because they wouldn't be doing a project that wasn't critical to their business. And so this idea of managing to timelines is really, really important. Bringing the right people to the party, and the expectations that professors would do this, that, you know, you would help us engage with the right people. And by the way, sometimes the right people are people you don't particularly care for. I didn't ask you if they were your friends or your relatives or anything else, or in your department or somebody else's department. What I want are the right people within not only this institution, but other institutions to help solve my problem. Money spent with universities is highly scrutinized. There's absolutely no doubt about it. So it makes delivering results even more important. It means delivering things on time even more important. It means delivering over expectation even more important. How about number 4? We all know it, right? There's almost always internal resistance when external experts are used. Every time you bring in a consultant, every time you bring in somebody from the outside, well, it is a good thing, and people realize that. There are also people that feel threatened because of it. You're questioning something that's been happened on the inside, and so there is resistance. The more that people have exposure to university, university people, to university administrators, to the people that work contracts, the better and the easier it gets. So the first time through is always harder. The more you do this, the more you work with groups of people, the better it is. Money spent is a metric for product importance. Money spent anywhere in business is a metric for how important they are, whether you're talking about the cost of people or the cost of materials or the cost of anything else. If the money is where the industry's vote is. And so if you have a $10,000 project, that may be a lot of money to a researcher here, but it looks a lot different than $1 million project within the organization. And oftentimes it has to do with the people that approve, the level that approves the expenditure of those monies. Every level within a company has some kind of spending authority level. And generally, it takes vice presidents or above to spend hundreds of thousands of dollars. And this idea that there are other people in the university you might want to connect with on projects beyond the people doing the projects. So some of the connections within projects at other universities have been up to the president's level, and that, putting the CEO and the president in a room can help, at least from an optics standpoint, from the way these projects are viewed, if nothing else. So how about if you're at the university? Many of the people listening to this are academics. Over-communicate technically, over-communicate about the project, over-communicate about, the possibilities, over-communicate, right? Expand the project, this idea of collaboration, real collaboration. The industry does not care about your department. It doesn't care about your college. It doesn't care that you like these people and don't like other people. It just wants the best answer. And so, and believe me, there are people in the industry that have a perspective on who should be involved. So there are people out there going, why isn't, Bob involved? Bob is a world-class economist. And the answer to that at the university is because we don't work very well with the economics department. But the industry guys are going, why are we using Bob? Bob was just on NBC this morning talking to, the host of the morning show. That's who we want to talk to. Schedule regular updates, and the more you can update the people in the industry above the project team, the better. Almost always, whether you like it or not, you're going to start projects small, and if you're patient, and if you're good, and if you deliver, sometimes, I did projects at another university, the Midwestern University. They started out to be $20,000 projects, $20,000 a year, and over a 5-year period, they went to $250,000 a year with the same professors, because we developed relationships that not only I trusted, but the people I worked for and the people that made decisions in the company became comfortable with. Understand the metrics. Do you really understand what the industry people are being judged on? What are they looking for is one thing. How this fits in with their performance metrics for the year or their bonus is really important. And then I would always under promise and over deliver instead of the opposite. Over deliver. It should be first on this piece of paper, but under promise and over deliver. Get my slide changer. So, you know, what are the keys to effective collaboration? We talked a little bit about what both sides, you know, how both sides think. There's clearly differences. There's a lot of things you can learn. None of this is unlearning. It's just things you have to learn. So the first is, you know, understanding how you're going to think about intellectual property and about collaboration in terms of intellectual collaboration is really important, because the best solutions aren't one party's or the other, they occur at the interface. And we all know that. And so you have to have an intellectual property agreement that allows, you know, people to share their very best ideas, knowing that they're going to, you know, be credited for that, rather than me giving my B grade ideas and keeping my A grade ideas for the, you know, FDA proposal or for my own, you know, patent rights or for a different project or something like that. So the interface is really where the power of all collaborations, but certainly the power between industry and academic collaborations occur. Multiple year projects are becoming harder and harder to justify. And so even, you know, the prevailing, you know, kind of perspective within industry is, even if it's a 3-year project, it's the contracts are 1 year, are 3 1-year contracts stacked. So you present a project for the first year, and assuming the results are good and the money is still in place, the industry money, then you proceed to year 2. But this idea of committing to multi-year project is growing less and less frequent. It's actually pretty commonplace when I started, you know, 25 or 30 years ago, but much less commonplace now. And with that comes this idea of breaking research goals into smaller goals with smaller price tags and shorter timelines. So rather than giving me one 3-year project, you go, listen, this 3-year project is really 3 projects or 6 projects. And again, I met people at K-State who are quite good at that. That makes the industry sell much easier. Trust is always the key. And the next one here, people that you may not have known. Some of my favorite collaborators and some of my closest friends were people who I walked up to at the end of presentations at academic meetings as an industry guy and go, I think you and I have similar interests. Is there a way that we could talk some more, that we get a cup of coffee, that I could come and visit your lab? And those relationships that started out with people who didn't know each other turned into relationships that still exist. One of those people I met 25 years ago, I talked to 4 days ago. So those are relationships that have stayed in place for a very, very long time, that started out with this mutual respect and personalities that worked and common interests. And that goes to the last, is there's benefits here that far extend beyond the funding that you would get or the things that are developed. And so again, there's some really good people on both sides, different goals perhaps, but really, really good people. So collaboration with multiple universities. It's a question I've been asked a lot. I've tried to execute this when I was in the industry. Some of my friends here, including Dr. Dorhout, putting, you know, multi-university agreements together, especially where there's intellectual property concern, is extremely difficult. And so there are consortiums that already exist. They've been built to minimize the complexity, but starting from scratch is challenging. It's not impossible, but getting things approved by different universities and then getting one industry or multiple industries to agree, again, especially when there's potential intellectual property, is just really, really difficult. And so there are associations that do this already, that fund research. Randy talked about that. There are other ways to get there, but trying to build this from scratch is time consuming and challenging. So, you know, kind of back to the beginning, you've heard in-depth technical conversations around food safety. It's an area, because of the Bill Maher incident, that I grew much more, much more comfortable with and much more aware of. I've done a lot of teaching myself. I think Randy would tell you that I'm a fair microbiologist, nothing to the level of the people who will or have talked in this call. But, you know, again, you learn what you have to. And so how does this play in the food safety world? You heard Dr. Abley talk about a little bit as regulations come out, and technology response to regulations is logical. Some companies are working on that beforehand. Some companies are more reactive. And so when it comes to the point where we might involve academia, then there's already a problem, and it's probably a substantial problem. So this is where there has to be a sense of urgency. The more you can be transparent in both sides. You know, I've talked to professors who said, well, it's hard to get things done, you know, between the breaks in the semesters because people go home, or, you know, even graduate students take a couple of weeks off. Those are things that you just need to tell. You know, all my students graduated and I'm going to have to hire new ones for this project. The more you can be transparent about that, the better. I would, especially in terms of food safety, regardless of what you thought at the beginning, design, plan, and execute those studies so that they could survive peer review. And even if the agreement is the industry has the option to publish or not, the data will be better reserved, you know, better absorbed within the company if it's just good, and if it's statistically meaningful, and if it's been planned well and thought well. And so this is one of the things I live by in my Sara Lee days and in my ConAgra days, that our work was good. Even if we decided not to publish it, it still was peer review quality, and it was designed so it could be published in a good, reputable journal. You always want to know that senior management in the industry is aligned with this. So it's one thing to have a junior scientist who's giving you a few $1,000s. It's different to have the head of a technical organization involved in the conversations. And, you know, the importance of the research, the money involved, the commitment on both parties certainly can be reflected by the level of management that's involved. Commitment to meeting timelines, and here's the academia, but it could be on both sides. But timelines are much more important because someone in the industry has told their boss, that you're going to know something by a certain date. And it doesn't have to be the answer, but it has to be, we have made the next step towards an answer, and here's where we are. And this idea of collaboration, the more you can involve the right people, the more that you can involve people from different perspectives, that's just a real positive. I think it's something we do decently at K-State, and it is certainly important to the way that people perceive that from the industry. So with that, I think that's what I have, Maureen. So are there any questions for me?
Well, we do have one sitting out here. In your former industry VP role, how did you critically evaluate university-based research outputs and how useful they were to your company? And what was a common red flag or two related to the university?
Yeah, that's a great question. That's another 30-minute.
You only have two.
That's another 30 minutes. Yeah, so that's terrific. So the answer to that is most projects are evaluated case by case, right? So you've committed to the deliverables. There's so much money on the table, and at the end of the year, there's an internal review team that takes a look at that project and makes some kind of decision on whether the money was well spent or not. after a few projects with the same university or with the same university partners, there starts to be an internal perspective or profile on, fill in the blank, on Dr. Phoebus. We know how he operates. This is the good, this is the bad. Or with K-State, we've worked with Dr. Phoebus and Dr. Smith and Dr. Featherstone and, all different departments. And we're seeing a common kind of way that K-State acts. And so that becomes, that coupled with the impact of alumni in management or alumni scientists starts to create this, halo, or it could be both positive and negative for academic organizations. And so there are actually people biased against some organizations, including some really good ones, and, and pro the organization as well. So it can work in both ways. Generally, in the organizations I've worked with who have substantial money, we would pick 4 or 5 universities that we had consistent success with, not only with the professors involved, but the people who are administrating the contracts and administrators and others. And those have become kind of our go-to for academic research. And we'd only kind of deviate from those 5 if we had a need that they couldn't meet, but they would be the first, they would get the first right of refusal in many cases.
Great. Well, thank you very much. That was a very, very interesting overview, and I think there's a lot of good information to consider as researchers move forward on trying to make some of these connections with the industry research levels. So thank you so much, Gordon, and I appreciate your time.
Yeah, thank you for having me. I've enjoyed it.
Okay. All right. Next up is, last but certainly not least, we've got Dr. Sally Davis, who's the Assistant Professor of Experimental Pathology, and Dr. Randy Phoebus, Professor of Animal Science and Industry, talking about the title is "It Requires a Transdisciplinary Team Tackling COVID-19 Threats to the Meat and Poultry Processing Industry." Now, this is based on a grant that this group recently received, and it was very exciting. I remember reading through it thinking how wonderful the approach and the topic was and how timely it is. So I'm looking forward to hearing more about that importance of interdisciplinary and where this project will take us. So Sally and Randy, the stage is yours.
Okay. Well, in the spirit of the Global Food Systems Initiative work and the mission, which is focused on the innovation, outreach, and also workforce aspects of our global food challenges, I'm really humbled that you invited us to come talk about our project at this point. We're still very early stage. So what Randy Phoebus, my co-PI, and I will talk about are how we actually pulled this project together and our future directions. To date, this has been an effort between the College of Ag and College of Vet Med with a little bit of input from the College of Arts and Sciences, but recently some follow-on work has also included the College of Engineering. So it is really becoming a university-wide effort that we are really excited about. The immediate project we'll be talking about in most of this presentation has 6 co-PIs, including Randy and myself and me, as well as Sara Gragg, who you heard from earlier, and Valentina Trinetta, both from Animal Sciences and Industry, and 2 faculty members, Harsha Thipareddy and Manpreet Singh, who are based at the University of Georgia. Specifically, they bring poultry strength to this project. With no further ado, I'm going to segue to my partner, Randy, so that he can set the stage for the problem space that we're operating in this proposed project. Go ahead, Randy. I'm going to switch slides.
Okay. Well, thank you. Can everyone hear me okay?
Yes.
Okay. Just so everyone on the workshop line knows, Sally's in one office in Vet Med and I'm in the other in Call Halls, and Sally's running the slideshow. So you may hear me say click. And I just wanted to point out this is great evidence of our interdisciplinary collaboration. It's just running the slideshow this morning. So my role in this presentation this morning is to give you a bit of a backdrop as to what's been going on in the meat and poultry industry, the food industry in general, as it relates to COVID-19 issues, and kind of give you a picture also of some of the things that have already happened or that are anticipated to happen. And then kind of lead that in as I hand it up back to Sally later on to go through the specifics of our particular funded research project. And there's one thing that I want to point out really early, that Sally is showing a picture here of a real meat and poultry processing facility, and you can see what's going on there. These people are close by. They're doing really heavy type of manufacturing work. You can see the dividers between these individual people because they're less than 6 feet apart. You can see that most of them have face shields and some have a mask on. So this is really not what the industry used to look like, say, last January. And so a lot of things are happening in the meat and poultry processing industry. But the purpose of this slide is to make it a point that COVID-19 is currently, and I don't think in the future, will be demonstrated to be a food safety hazard. So it's not something that we believe or anticipate being transmitted through our meat or food supply. But it is a personnel hazard. The health and safety of the actual workers in the plant is what we're focusing on. Now, with that said, and I have a later slide to go into this just a little bit more, when something of the significance of COVID and the reaction of the individual plants to controlling COVID happens, obviously these actions, these foreign type actions that we're doing in the plant can have food safety implications. And I'll point out some of those as we go through these next couple of slides. Click. Okay, just a little bit of background here. Click one more time, Sally. This is the national picture as we have it today. Actually, this was yesterday on the New York Times website. This is not looking specifically at the meat and poultry industry. This is all of society. And as you can look and see here, about mid-March, we start kind of seeing COVID cases nationally. Were those cases happening before that? Probably. We weren't testing. We didn't have testing measures. We, you know, the kits and things that we now have. But I'll tell you right now, I was sitting in a hospital room with my very ill father at that particular time, and I saw what was happening in the city of Nashville, Tennessee, which is a large city, and what was happening within the hospitals. There was a lot of preparatory action going on and a lot of angst about what was going to happen. And, you know, this is nationally. So you can kind of follow this graph, New York Times graph, and through the end of June, we were ticking along at about 30,000 cases a day. This is a rolling 7-day average, but it's reported on a per day basis. So about 30,000 nationwide. And then between June and mid-July, we see a pretty good significant spike nationally, where we're up between 50 and 60,000 cases per day. Through the next few weeks, those numbers started coming down just a little bit. But as you can see, beginning, say, in October last month, nationally, our cases are going back up again. And you know, this is we can speculate all we want on why this graph looks the way it does. Yes, we've started testing asymptomatic carriers. We're testing with better equipment. We're testing a lot more people. So, you know, kind of ferreting out what really this graph is telling you is hard. But I will say, when you look at reports coming out of, for instance, Wichita yesterday and this morning, we are seeing much higher numbers of cases presenting themselves, and hospital beds are filling up in cities like Wichita. And so this is a concerning issue for society, but it's also a concerning issue in food processing, particularly meat and poultry processing. Sally, can you click? Okay, this box, this was a snapshot in time that was taken back in early July at looking at meat processing plants. This is CDC reporting here, and they kind of did a survey, and it was revealed that there was over 16,000 cases, infections in meat and poultry processing workers during this time, and they had 86 deaths. But there's some problems with this data in that only 28 states had reported, and that only covered 118 plants. We have about 9,000, over 9,000 plants in the country. And when you looked at the numbers of cases reported per plant, it ranged anywhere from a few, for instance, 51, all the way up to 1,000 or more cases per plant. Now, this was in July, kind of in the height of the situation. I will tell you from personal communications with the industry personnel that these counts in the industry have started coming down, actually, at the same time that counts in the general population are still going up. So to me, that indicates that the industry, the meat and poultry industry, has really done some yeoman's work at addressing this. Now, do we still have issues? Absolutely, we do. And that's part of the reason that Sally is going to tell you about this grant that we were awarded. Okay, Sally, next click. This timeline on the bottom, and this only goes through May because I wanted to point out some things here. Back in January and February, COVID started hitting our country and hitting the world, really, and no one knew hardly anything about it, correct? It was kind of blindsided about this. We didn't know what it was. We didn't know who it infected. We didn't know any kind of way to treat people or isolate people or whatever else. And, the same thing happened within the meat processing facilities also, but kind of at an amplified way. Because if you think about these meat and poultry processing companies, and I'm going to give you a little bit of for the people not familiar with such operations, kind of a visual of what it would look like. But I've thought myself over the last couple months, if I had to pick a scenario that was probably the worst case scenario for COVID-19 infection in our country or in the world, I would have probably picked a meat and poultry processing facility. And the reason is because we have so many people working side by side in very non-distant manners. We're doing a huge amount of activity, a huge amount of airflow that's going on. And we've got some really unique issues that are unknowns for us. And this is what Sally will tell you we're going to be addressing in our work. What's the impact of chilled and high humidity conditions or humid conditions on a slaughter floor that are more at 80 degrees Fahrenheit versus conditions on a fabrication floor that are 4 degrees Celsius, or let's just say 40 degrees Fahrenheit with very low humidity. Even some of our work that's already been accomplished here, ours being K-State's work by Dr. Jürgen Richt and his group, have shown that this virus has very different persistence characteristics when you're in those different environments. So again, the meat and poultry industry really got hit hard. And along this map, you can see when we first started, for instance, CDC was saying no mask back in February, encouraging people not to wear masks, as a matter of fact. And then all of a sudden, we get the announcement that we need to wear masks. Well, you can think about what that would do to a huge processing plant when you're having to make that change basically overnight. But something really important happened early in the March timeframe, is we realized, or we formally declared, that the food system is a critical infrastructure in this country. And we can't just shut it down, the manufacturing sector. And shortly after that recognition was made, and you'll see one more click, Sally, over toward the May timeframe, the Defense DPA Act was executed to ensure that our food systems, but particularly our meat and poultry systems, stayed open. So this Defense Protection Act was an executive order from the president, and immediately our Secretary of Ag sent out letters to governors and CEOs of 50-something different meat manufacturing companies, asking them or telling them to stay open and stay producing food, keep producing food. Well, that was an order that they had to comply with, but they also had the obligation to look out for the welfare, the public health of their employees and the communities that they lived in. So they were really in a precarious situation to be able to respond to the DPA Act, and they did. And Sally, let's go to the next slide, if you will. This is another graph that USDA put out. Looking at blue lines are the number of cattle killed weekly, and the red line is the number of hogs killed weekly. And as you can see, through the first part of the year up until probably the end of March, we were ticking along at normal. We were killing about 65,000 pigs per week, hogs per week, and about 70,000 cattle per week. And then all of a sudden, we get into this period of time where a lot of the illnesses and outbreaks were happening in the meat and poultry facilities. We had workers becoming ill, and I saw some statistics where 30% or more of employees at the plant were carrying, whether it was symptomatically or asymptomatically, the virus. And so with that happening and these plants shutting down, we ended up with a real bad scenario on our hands as far as our meat and poultry supply chain. So, you got your livestock producers who have fed these animals out. You've got 1,200 to 1,400 pound cattle in a feedlot that have to go to the processing facility, but the processing facilities were shut down. If you look at what was going on, particularly up in Iowa with hog slaughtering, we had producers who had nowhere to go with their hogs. They and the USDA were preparing plans to depopulate and dispose of large numbers of animals. And fortunately, we were able, through just a mammoth effort, to turn that around in May, in early June, so that we could open these plants back up and serve capacity of keeping that supply chain active and going. Click, Sally. So the picture here depicts the buildup or the backlog of animals, whether it's poultry, cattle, or pigs. Click. And the impact that had on the meat supply, that I'm sure everyone can remember going in May, maybe, to the grocery stores and seeing food shortages. Yeah, I don't know what was worse, not being able to buy a steak or not being able to buy toilet paper, but we had both going on at the same time. And so it's not just meat. We had Gordon over in Grain Science knows that there were shortages of flour and rice and everything else on the market shelves. But not only were there shortages, that led to very significant increases in price of the product that was available. And so most of us saw what was going on at the supermarkets, but my daughter works in a restaurant industry, and even when they started reopening to some level, that was delayed because of shortages of retail ready product that these restaurants needed to start back operations. So it was a really revealing incident as to what our supply chain is, and how, I guess, critical it is, but also how fragile it is in some cases as we think about it. Now, with the green circle that I'm showing here, I heard Dr. Tonzer, one of our K-State ag economists yesterday, talking about the reduction in our slaughter and product capacity was about 40% during this time. So just imagine in a matter of a couple of weeks, reducing your slaughter output by 40%. Obviously that is going to impact prices and supply. But I also want to show you the far end of this graph, which is now, you could carry that on out further now, but we have basically been successful in opening these plants back up so that we are now again producing, slaughtering and producing at pre-pandemic levels. What happens if we have a second wave? Well, who knows, but with all the infrastructure and the programs and the learning that we have had over the last couple of months, most of the economists, including Dr. Tonzer, doesn't believe that even in a second wave, we would see the dip that we saw during this first wave. Okay, next slide. Sally, there we go. This slide is, one more click if you mind. This slide is kind of profiling or summarizing the great response that the meat and poultry industry has made to trying to rectify the health issues and the plant shutdown issues in the facilities. Again, I wanted to kind of give you the vision in case you're not greatly associated with the meat and poultry industry. Just think about an enormous facility that is a big industrial complex, and you've got thousands of workers working double shift in this plant. So you got social areas like locker rooms and hospital clinics and all kinds of things inside the plant. But you've also got all of the harvesting operations and the fabrication operations. So you got multiples of thousands of people on those processing lines, shoulder to shoulder, doing very heavy work. And then you've got also all of the contractors, which could be hundreds of contractors coming in to handle HVAC systems and things like that. So that comes back to the point I made, this being a very, very complex thing to try to deal with an agent like COVID in. So that happens. And then you've got all of the sanitation shift, which is normally a third shift company that comes in with a couple hundred workers and cleans things top to bottom. Well, COVID, SARS-CoV-2, we don't have, or we, at the time, didn't have a lot of information on the effectiveness of cleaning protocols. And most of our chemicals weren't specifically approved as control agents for SARS. We had to use other viruses that they had been tested against and things like that. So now we have gone through this period of time where we've done a lot of personnel testing, almost daily, 100% of workers getting tested. In some cases, instead of shutting plants, they were able to slow production lines when they didn't have enough workers. As that very first picture I showed you with the barriers and things, a lot of those things have been constructed that prevent aerosols from workers side by side. The left upside down triangle here is put out by the National Institute of Occupational Safety and Health and CDC. And this is, they call it a hierarchy of control. And this is what Sally and our team that's working on this is kind of following. Yes, we have PPE like masks and things like that that everybody's using. The industry's doing a lot of the administrative control aspect of things, changing the way people work. How do they go to breaks? How do they go to the cafeterias for lunch in their locker rooms? How often are they tested? Things like that. One of the big areas that a slide here next is going to show you that we're starting to work in is this engineering control aspect. What can we do with these companies to design physical systems that reduce the risk to employees? And also, you know, at the very top of the box is elimination. And yes, we have the goal of eliminating this. Maybe that is this being the virus. Maybe that is through, you know, UV light or other things that might decon air or filter air. or maybe it is our ability to manage the virus once we have a vaccine and we can vaccinate these critical employees. So we're working in this hierarchy control where these boxes, if Sally will click one more time, we're working at all of those areas. But you know, there's a lot left to learn. We are at a point where we are gaining information relative to food manufacturing, meat and poultry manufacturing. There's a lot of information accumulating out there in the ether about how the virus, how long it persists on cardboard and, you know, in hospital rooms and school rooms and things like that. So some of that we try to translate to what we're going to be doing, but others, you know, our industry is so unique that we either have to develop new information or verify that what we see elsewhere is valid in the meat and poultry industry also. Click, Sally. So I'm gonna do these one at a time, but Sally, you can just go ahead and click all the way through there if you want to, so I don't have to say it so many times. If we look at the future, you know, from where we are right now to the future, there's quite a few things. First of all, and our grant, as Sally will tell you in a minute, is going to try to address most of these. First of all, is we need to have a science-based management plan, or the companies do, and we need to be able to help them with the data they need to make that plan. We have to really think about and anticipate a second wave of COVID-19. From the discussions I have been having with the industry leaders, they are anticipating a second wave of COVID as more people are coming indoors, either in the community or at the plant. You know, now they're sending people outside to eat lunch. Now they're going to be packing the lunchrooms a little bit tighter. So they expect that. But one other thing, and Dr. Davis is one of our very knowledgeable people on campus about influenza. This is the time of year that these meat plants also have to deal with human influenza in these same settings. So how does that overlay over COVID? The government and customer COVID control requirements, particularly the government, have put out guidelines, and these companies are being challenged to follow, implement all these guidelines, even though some of the guidelines are not really proven. They're just kind of logic type things. And, you know, there's right now a lot of lawsuits and a lot of OSHA fines happening that are putting pressure on these meat manufacturing companies also. And that's going to carry out probably for the next 2 or 3 years. If you have a line worker, loved one who dies from COVID, was it the company's fault that they stayed open and kept manufacturing because the president and government told them to, or is that company liable? So those are some of the things. But in that box at the very bottom, or the under the arrow, I just wanted to kind of circle back as what I said earlier. COVID is really not a food safety issue. But if you look at food safety that these companies have to deal with day in and day out, we are refocusing operational budgets, for instance. All of these modifications and these testing programs and all of the fines and lawsuits are taking money away from the processors to deal with COVID that could be going toward food safety and food quality. We have reduced food safety personnel, people sick, some people are afraid to come into work, and so that poses a challenge. We have reassignment of some of the food safety and quality assurance personnel to focus on COVID control. One of the vice presidents in one of the largest meat companies who normally does food safety, she has been put into a COVID management program for a multi-facility international meat processing organization. So all of those things could impact food safety, along with, you know, our grant is going to focus a lot on cleaning and sanitation. And if we do better in that area, including personal hygiene to control COVID, my speculation is that we will make improvements with other normal food safety hazards. So that I'm kind of looking forward to that happening if it does. And that would also include the engineering controls like a better air quality and things like that. So, the final bullet I put here is, and I want to be real careful with this, is, right now we do not see any evidence that SARS-CoV-2 is a foodborne pathogen. Now, we might have to change our mind at some point down the line as science presents itself. But right now, I think we're pretty solid in making that call. Click. Okay, so this will be, I think, my last slide. Look at this in terms of the overall system here. We have our national community, we could say international community. And within the processing plants, if you kind of follow this around, going down first, so these manufacturing facilities are in local communities. And so what's going on in the meat plants can definitely spread to the local community. And we have seen this in places like Garden City, Kansas, and place where these companies are. We have the plants themselves and all of the things that happens exterior to the physical facility, but then you move inside to the direct meat and poultry processing area. And that, Sally, if you'll click, oh, okay, sorry about that. This is where we are focusing most of our effort at this point in time, is where we are processing meat and harvesting animals. That's what our grant's going to be about. But you can also see these employee social areas, such as locker rooms and cafeterias. We are definitely interested, and as a matter of fact, outside of the COVID grant that Sally's going to speak about that we got from USDA, we are working with companies and technology providers to expand our focus, our emphasis on some of these areas too, because they're just as important in our eyes. Okay, Sally.
Okay, thank you very much, Randy. Got a laser pointer now, apparently, so we're good to go. So I'm going to transition now to what came across my desk on April 22nd or thereabouts, which was the announcement from the National Institute of Food and Agriculture at the USDA of the rapid response research on COVID-19 impacts on agriculture. And so per usual, I was looking for opportunities to apply for money to fund research. And this came across my desk, and it was focused on food across the food and agricultural supply chains. So this is a very broad grant looking at the agricultural systems themselves, food supply, and the families and workers impacted by SARS-CoV-2 and education regarding that. It had twin goals of workforce, a healthy workforce and a safe food supply. And it ended up being an amendment to the Agricultural and Food Research Initiative, specifically the Foundational and Applied Research Program, which is a competitive grants program that I had actually applied to before in a different context. However, when I looked at the priority areas from my seat as a virologist, I noticed that I would have big gaps in my knowledge and my discipline expertise to actually respond to this, and that this was going to have to be an effort that involved multiple disciplines. Specifically of interest to me was the food and food processing piece. And at that point, I remembered meeting Randy Phoebus and our joint interests in containment research, with his being in the microbiology side and food safety, and mine being over in the virology side. And we've met in passing related to some Biosecurity Research Institute event in the past. So I took this information, the call, and I sent it to Randy and asked him if he'd be interested in putting something together. So specifically, as Randy talked about earlier, he's particularly interested in the meat and poultry processing field, and that seemed to be the one that was in the most pain. And if you look at these 5 objectives that came under the food and food processing section, after reviewing them together, we determined that all 5 of them were relevant. So, for example, determining the survival or the amount of infectious SARS-CoV-2 on various surfaces applied both within the food and the manufacturing environment as well as to the food itself. And we were particularly concerned about the exposure of workers within that environment that Randy just illustrated to you. To assess the situation where the actual conditions themselves, which are really unique, impacted this infectious virus was of the next concern. And what's particularly important to point out here, alluding to something Randy's already mentioned, there was quite a bit of literature that was already out at this point and forthcoming related to how well the virus lasted, its persistence on various surfaces. But most of the surfaces that were the subject of those research articles were clean surfaces. They were paper, metal, surfaces within the hospital environment. And even when we start talking about management or elimination of the virus, once again, set within the hospital environments, particularly as regards the disinfectants improved in those environments. So what was missing were a consideration of the unique environmental conditions that Randy's referred to in the meat and poultry processing environment. Not only do we have high humidity and high heat in the slaughter side, but we have an incredibly cold environment, as well as, in some cases, areas of desiccation within the actual packaging side. And we end up moving between these environments with the same piece of meat in its journey through the supply chain. And throughout that whole process, we have workers. We have meat, which is not a substance that when it hits a surface doesn't leave a residue, passing over lots of different surfaces, plastics, stainless steel, all sorts of different types of contact with various PPE that was already extant in the industry, and now PPE that had been added to the industry as we tried to protect these workers. And we lacked an understanding of where the virus lurked and remained in that environment. We also, as Randy pointed out, had very little understanding regarding the extant USDA disinfectants and sanitation approaches. We thought that they were likely very efficacious, but we had no actual proof with this virus itself. And recent research has shown that there are some interesting characteristics of this virus that were non-intuitive initially. For example, it was assumed initially that it would survive better in a humid environment, and actually recent research says the opposite. So we needed to look at the context of the meat and poultry processing environment as regards where and when this virus remained, and also what that was already in place, already USDA improved, was working. And the second step then would be what could be modified within existing USDA guidelines from FISIS, and if there were gaps, then to work with the industry to address those with the government. Finally, as is true to our extension mission here at K-State, we felt firmly that we couldn't just go off and do all this research in a bubble and publish some primary papers. We need to integrate from the beginning with the people who will benefit from this research, specifically the meat and poultry industry itself, develop logical education materials along the way. So we thought of all of this, these objectives, and that's what we came up with in our early discussions. And we also concluded that this was definitely something we wanted to go for, but that we also would have to bring a very interesting, unique team together with multiple disciplines. So again, we focused in this direct MMP processing for this specific opportunity from the USDA. So I'm going to take a step back now and just get us all on the same page with some definitions and some terms. When I refer to discipline, I'm talking about a branch of knowledge. Typically, it's one study in higher education, but not always. The first discipline that came to my mind when I was looking at this call was virology. It's an obvious one, but I'd like to point out a few aspects of it just to ground everybody together and talk about some relevance of the particular call. So virology is the study of viruses. In itself, it's an interesting field because the subjects are not visible in most cases through a light microscope. We can't see them. Additionally, viruses aren't technically alive. Gets into the whole concept of jargon around them and a bit of confusion about whether we're dealing with a living thing or not. they're not alive until they enter a cell being, in this case, for SARS-CoV-2, a cell in your respiratory tree in your lungs. So in this case, they're kind of hard to wrap your head around, and they're certainly impossible to see without other techniques involved. And regardless of their presence in the environment, they can sit in the environment in an abiotic state. And so long as they're intact, they're potentially infectious. So what we wanted to look at was what SARS-CoV-2 virus was still extant in this environment. Was it being eliminated by the third shift, the sanitation shift? And also during an average workday, where was it landing? Where could it potentially cause problems? And how could we mitigate that? We also discovered early, as we pulled this team together, that Jeanette Thurston, who's the head of the Food Sciences Institute, has a background in environmental virology, which was highly complementary to my background in more tissue and animal-based virology, with my background in veterinary science. And so that was brought into the team to consider all aspects of where the virus might be. So as Randy mentioned, my background is primarily in respiratory viruses, specifically influenza A, as well as some other emerging and zoonotic viruses, mainly animal-derived. However, since SARS-CoV-2 is a respiratory virus, I feel at least at home in thinking about its pathology. The closest relatives of SARS-CoV-2, as you might know from the press, are SARS-CoV-1, renamed 1 after the discovery of the recent virus. There were outbreaks in a pandemic in 2003. It was very scary, but fortunately very short-lived. Its other near relative is the Middle Eastern respiratory syndrome virus, which causes sporadic outbreaks, mainly in the Middle East, and is actually a much more deadly virus than the first SARS. Despite my background, I knew that the team would be immediately strengthened by someone who is an expert in coronaviruses. And we're very fortunate to have Dr. Yoonjung Kim here at K-State. And so we approached her to join the team because every virus, every family of virus lends itself to some interesting idiosyncrasies in terms of how we grow it, how we look at it, and how we measure it. So she joined our team quite early. It also occurred to me that in speaking with Randy, that virology, unlike microbiology, is a hell of a lot less rapid. It usually requires several days to measure a result because we have to grow cells, and the virus itself takes less time. So I started thinking, well, that's not going to be very helpful if we are low throughput and we have a situation where we can't be rapid enough and put a large enough set of samples through to be rapid. So I started thinking about that and started thinking about some aspects of technology. We didn't want to default to what a lot of the literature is doing, which is only looking for presence of a virus or its footprint in the sense of its nucleic acid, which actually doesn't indicate whether you have infectious virus. But we wanted to find a way to assess the presence of potentially infectious virus in a much more swift fashion. Another important point of this project then was to think about this within the context that Randy's already brought up, the public health context, which I firmly agree with Randy is the main point here, not food safety. And that's the health and human safety side of it. Now this suffuses the project. We don't have a specific expert on this, but various members of the team have a background, and multiple faculty on the project actually are involved with the Masters of Public Health here at K-State, which in itself is a wonderful pan-university effort. So we have public health in mind at all times while we work through this. Then we got to containment science. And by containment science, I mean for us using the Biosecurity Research Institute, which was focused on earlier by Peter Dorhart and mentioned later by Sara Gragg. It's a fabulous facility that offers us the ability here at K-State to do biosafety level 3 work. That is a requirement for SARS-CoV-2. So one of the things that we had to think about within this project was, did we want to use actual SARS-CoV-2 virus, or did we want to use a near relative that would allow us to do it work outside the BRI? Our answer to that was actually we're doing both. We're doing methods development at BSL-2 with human cold viruses, which are also near relative coronaviruses. And we saw as a key differentiator for our work the use of the BRI. More so, leaning on Randy and his experiences at the BRI, we are leveraging the excellent meat and poultry processing efficacy of equipment over in the BRI. We have industry standard equipment, very similar to what you saw in the images earlier at the BRI, and it's proven in its use in prior projects within containment. So those were key differentiators on the proposal, and our team itself, fortunately also at this point, with the addition of Sara and Valentina, and Randy's graduate student Danielle, as well as Aaron Schertzinger, who is now a postdoc on the virology side of the project, and me, we had quite a bit of work within the BRI and prior experience we could point to. So this brings us back to the meat and poultry processing domain expertise, which is an obvious critical on this project. And I also want to say take a moment to talk about our fifth discipline, which is outreach. As mentioned earlier, we could have just proposed a research project, but we felt that we would fall short if we did so. If we created just another body of research, some information on surfaces within the meat and poultry processing environment and the longevity of SARS-CoV-2 there, we wouldn't be meeting our objective, which was to actually help industry partners. Multiple members of the team have strong relationships with many different meat and poultry processing companies, and they were very acutely aware of their pain. The outreach team, therefore, looked at this in a holistic manner, and we brought in poultry expertise from UGA at this point, as well to complement the cattle and pork expertise here already at K-State. And by outreach, we mean everything from 1-pagers to 2 to 3-page guideline descriptions to working with industry to gather from them inputs such that the outreach we make for them is more specific to their needs. And as I'll get to in a moment, we discussed actually doing formal modeling, mathematical modeling of the meat and poultry processing environment and taking data inputs from extant literature as well as our research itself as inputs into these models so that we could do risk assessments that were relevant to the industry. So we have a lot of disciplines here, and I was beginning to get overwhelmed as the lead proposal writer. So how do we go about moving through this? One of the things I'd like to point out is there's a lot of these something disciplinary words bouncing around out there. These are the definitions I use. Multidisciplinary is combining several academic or professional specializations to approach a problem. They're not necessarily integrated as they would be in an interdisciplinary project where you're analyzing and synthesizing across a whole using multiple disciplines. What we strive for is to be transdisciplinary by integrating our different disciplines, getting rid of the jargon, which has been an interesting find in this whole process for me, or at least learning how to speak each other's language and transcending these barriers in order to perform as a team and develop in itself a new discipline to address the need of this industry. So in summary, multidisciplinary is additive, interdisciplinary is interactive, and transdisciplinary is holistic. So pulling those disciplines together that I showed you before, we wanted them all to influence and be involved in this whole, which was the SARS-CoV-2 project in the meat and poultry processing environment. This was going to require communication. I mentioned the jargon, but also just communication to keep everyone on board with the vision and then the actual implementation of that vision. Project management flows from that naturally. And then back to both the global food systems push for workforce development and our own personal interests being academics who really cared about the next generation. We wanted to do training at all levels, not just the training of the outreach type, but within and without, within our team as well, in terms of training at different levels of students as well as postdoctoral training. So here's our research team. I've mentioned quite a few of them. As many things in the current pandemic situation, not all of us were able to make the photo call. In fact, 2 people were under quarantine at the time and the rest were remote. But Pictured here are Daniel Vega, who is Randy Phoebus's PhD student, a senior PhD student who we will work to transition to postdoctoral fellow status during the lifetime of the project. Valentina Trinetta, that's me, Randy and Sarah, who I've mentioned. Erin Schertzinger is not pictured, but she's a senior postdoc who has switched from College of Arts and Sciences to College of Vet Med and now is fully on this project. Yunjin Kim, who I mentioned, who's critical on her coronavirus knowledge. Anka Richter, who I haven't mentioned in name, critically joined our project to handle mathematical modeling. Anka's at the Naval Postgraduate Academy. She specifically has a research mandate of public health operations research, which means her focus is mathematical modeling and risk assessment for public health, more specifically an interest in infectious disease. And so she has a long track record in that field. Manpreet Singh and Harsha Thipareddy are our UGA folks, and we're in the process of recruiting 2 master's students. So if you're a master's student, you're looking for a new opportunity starting in January, we do have advertisements up for graduate research assistant positions on this project. I realize that we're running a little late on time, Maureen, so please chime in and let me know how much more time we have, because I believe I started.
Yeah. Why don't you take just a few more minutes and then we can wrap it up. This is great.
So specifically, this is our project goal as presented to the USDA. We wanted to identify, develop, validate, and deliver, meaning the outreach side, practical cleaning and disinfection strategies. The quantitative risk model piece is also very significant because we want to be able to do science-based decision-making with our industry partners. The SARS-CoV-2 persistence I went into in quite a bit of detail, and the effectiveness of the existing USDA-approved cleaners and sanitizers, really relevant because of how this industry is regulated. And finally, we wanted all of this to come out to the industry in a timely fashion. We have an executive management team. These people I've already mentioned. For some reason, Jeanette missed the roll call on the picture. I'm sorry, Jeanette. And then these are the institutions. Emma Moran is our program officer at the USDA. Critical to this project is the Industry Advisory Board. We have 10, we won't name them for their consideration, large multi-facility meat and poultry companies. They are including ones that are further downstream, large retail, such that they actually package or rehandle supplements. So we have an incredible group of people across all aspects of operations, not just food safety, but other aspects, including financials and risk management, and specifically some people who have been tasked to look at COVID-19 within these environments. And finally, our information dissemination will go from the red of UGA to the blue of K-State. And NC State has a framework in place that we'll be using that is across all the NEFA projects, and we'll be using that for dissemination of information too. I'm going to skip these objectives. I can go into those in the questions of specific points you want to be looked at. We do have this 1 million. One other thing I would want to mention besides the 1 million from the USDA, which we successfully secured, we realized that this project would be very expensive considering the BRI rents, the fact we were repurposing a microbiology lab in the BRI to do virology as well, and the fact that we were going to add some technology. So we're really thankful for the support of the state of Kansas in the form of the NBAF fund. They matched it 1/3 of our project costs. These are some of the technologies we used. And so that then added the need to work with vendors, including vendor development teams, to our transdisciplinary project. I'm going to switch back to Randy. He has one point to say about the employee social areas and the introduction of engineering, and then we're done.
Okay, Sally, thank you so much. And I went too long, not Sally, so I apologize for that. But just coming back to this slide that we put up, and I've already mentioned this, but the USDA grant is focusing heavily on that red box there on the direct meat and poultry. As I mentioned, those employee social areas, and here we're talking about everything from locker rooms to cafeterias to religious areas, and those are really important too. And So we have identified several technology providers, air sampling companies, along with the meat companies themselves on engineering new technologies that would pull that virus away from personnel or filter it out or kill it. And we think that this is just going to be not only a next 1 or 2 year project, but I think we may be able to develop a whole program around this part of the food manufacturing sector. And obviously our findings in the food sector will transmit to other places like schools and hospitals or whatever. So that's all I really wanted to say about this final one, Sally.
Okay. Do you need that final slide, or should we
No,
just?
I think we're good.
All right, thank you.
All right, we have gone over a little, but I think that the information was well worth the extra time here. One thing I do want to thank you for, Sally, is the description of the disciplinaries. I tend to use the word multidisciplinary frequently, and it's kind of a dumping ground for all three of those activities. And the transdisciplinary is so critically important in some areas, and it's not easy to do. And the way you've described it and talked about the challenges with language and with being able to communicate easily between these groups is, I've heard so many times with groups that have tried to do this. And I commend you for the work that you guys have done, and am really excited about this project moving forward. So thank you so much for the overview. I don't see any questions coming in specifically at this time, but I'd like to open it just for 1 or 2 minutes to see if any of the speakers have comments or questions that you would like to present to one another. I don't know if Melanie is still on, if she's got any input here, or if you've got something you would like to ask one another, any comments?
I'm still here, but I don't have any questions at this time.
Okay, very good. Well, with that, what I'd like to do now is just thank all of you so much for your time, and thank you for the great presentations that you've put together. The Global Food Systems Initiative at K-State is better off for the efforts that you've made here in trying to get the word out. I will continue to reach out to faculty members, and certainly if you've got any thoughts or ideas of ways that we can get more information out between your colleagues and off campus to others, to more folks at the USDA or wherever it might be, but I'm all ears. So again, thank you so much for your time, for your efforts here. Thank you for those that have been listening in. We will be posting this out on our website. So if some of your colleagues were not able to join us today, we will have it out on our website, and you'll be able to listen to all of these presentations in the future. So again, with that, we'll go ahead and sign off, and hope you have a wonderful day.